A very good afternoon to all. I welcome you all again in afternoon session of second day of our national webinar that is application of biotechnological tools in crop improvement. So as per schedule, the first lecture will be delivered by Dr. C. Tara Satyavati Madam and then Dr. P.M. Kaur sir. Now I will request Chairman of the session, Dr. A.K. Singh sir, Director Instruction Service KVP Gwalior, to welcome ma'am, welcome the speaker and to deliver their talk. Thank you very much.
Good afternoon, madam.
Good afternoon.
This is the second day session, an afternoon session, and there is two reporters, Dr. Akhilesh Singh and Dr. Amit Bhadoria. And now I introduce yourself to the viewers of this second day seminar. Dr. C. Tara Satyavati has completed his PhD degree in 1998 from IRI New Delhi, and presently she is working project coordinator, All India Coordinated Permanent Improvement Project Jodhpur. In the past, she was principal scientist at IRI, and the major research contribution in the quality parameters in rice, traceability relationship, and with hybrid in Basca, genetic improvement, development of minimum descriptors, national test guidelines in reference to varieties for dust testing in soybean, identification of TTL for seed port permeability, breeding for both seeded soybean. Seed-oriented breeding program at IRI developed one hybrid, three varieties, identified TTL for DM resistance and agronomical important traits. He developed high gain FEG wing fulminate lines, and besides this, uh, SPC, he introduced minimum FEG content in promotion criteria in the variety release policy first time in the world. And she got so many awards like Punjab Rao Deshmukh Outstanding Women's Award 2016, Outstanding Millet Scientist Award 2018, and Dr. M.S. Swaminathan Women Scientist Award 2019. She has published about 58 research papers in national and international reviews, 6 books, and handled about 20 research projects financed by different agencies, and she has guided 3 PhD and 8 MSc students. She is very intelligent and outstanding lady in the field of formulate. I welcome you on the behalf of RVS Warrior. Yes, madam, please start your celebrations. Okay.
Thank you, Dr. A.K. Singh, for your nice words and introduction. A very good afternoon to all of you. And I thank the organizers for giving me this opportunity to share my experiences and the work taken up related to biofortification in pearl millet. So when I was asked to give a topic, I just thought, because this webinar is addressing the students across the country, both the graduate, undergraduate, and postgraduate students. So I thought, let me try to give them a feel of what biofortification is and what we are doing in pearl millet related to biofortification. So my presentation would be divided into different parts. Firstly, I would like to take you along with me and tell about biofortification, then what happened in different crops, and then what is happening in pearl millet, and then what are the things we need to do in this COVID-19 scenario. So here I go. So if you see the world population, it is constantly increasing starting from 1950. Here I'm showing till 2050. And there is a constant increase. And the pressure on agriculture is on the farmers and different nations is to produce sufficient quantity of food material to this ever-growing, ever-burgeoning population. Now, apart feeding them, we also have to see that they are given quality food. And if you see the statistics that there is global micronutrient deficiency mainly related to vitamin A, zinc, and iron. And this kind of deficiency is most prevalent in the southern hemisphere, especially in case of Africa, South Asia, and South American continents. And if you see why we should talk about these nutrients, what is their role? We should know what are the roles of these different elements in our human body. If you see there are 51 essential nutrients to sustain human life, of which air, water, and energy, these are very, very important, without which we cannot sustain. Then protein, that is in terms of amino acid, 9 amino acids like histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine. These are important. Then lipids like linoleic and linolenic acid, fatty acids. Then minerals, macrominerals which are needed in a large quantity like sodium, potassium, calcium, magnesium, sulfur, phosphorus, fluorine. Whereas microelements or trace elements, these are iron, zinc, iodine, copper, manganese, all these things. These are 17 in number. Of these, today I would focus on iron and zinc with the relation to pearl millet. And then vitamins, there are 13 vitamins. Deficiency of any of these nutrients, 51 essential nutrients, will reflect on our system, either in some problem or health issue or anything. So we should see that whatever we take, it should have a blend of all these things. Now, if you say this is a malnutrition poverty trap. Now, these are all unending, interlinked things. Because of poverty means that is low earnings, people are not able to get good food, low productivity, and then it ends up in malnutrition. And malnutrition it could be because if from one side poverty is leading to malnutrition, but on the other side, even though people are rich because of the poor diet, it is leading to malnutrition. Now, globally, 1 in every 3 people are found to be malnourished. And the global crisis, this particular global crisis is happening now. And what are the consequences of this micronutrient malnutrition? Higher morbidity, higher mortality, lower cognitive ability, lower work productivity, impaired growth, and impaired reproduction. Nothing is left out. And it was also highlighted by Harvest Plus that in India, vitamin A deficiency supplements reduce child mortality by 23%. So, if you are taking any supplements to address this issue, child mortality has reduced by 23%. Otherwise, 3,75,000 children are going blind each year because of this vitamin A deficiency. Iron deficiency because of this impaired cognitive abilities, which cannot be reversed back. Once you lost it, you are lost. So 82% of children less than 2 years in India are anemic. So we should see how important it is. Zinc deficiency, increased incidence, severity in diarrhea, pneumonia, stenting. These are caused by zinc deficiency. Nearly 2 million people are affected by this. So if you see how important, though these nutrients are required in small quantities, very minute quantities, but their impact is very significant on our health. The deficiency, the malnutrition because of all these things, generally termed as hidden hunger, we can see the impact in the life cycle in all the stages, starting from newborns. If you see the impact of hidden hunger in newborns, it results in low birth weight, high mortality, reduced mental development, and makes them often ill. Whereas in children, they are stunted in growth, they fall often ill, they are small in stature, high mortality, and low resilience. In adolescents, they're stunted, reduced mental and physical performance, and they're always ill. In pregnant ladies, high mortality, frequent perinatal complications, and lower performance. Whereas in adults, poor, malnourished, and limited productivity. In seniors, it leads to often chronically ill and lower life expectancy. So, if you see, none of the stages in our human life are unaffected with this. So, at each and every stage of our life, we have to see that we should have proper quantity and balanced amount of these nutrients in our diets. Now, how do we get this? Some people say that okay, if I'm deficient in this, I can go and take some food supplements. Okay, food supplements, if we are ready to use food supplements, we have to pay for that because we have to buy them in the medical stores or the pharmacies. Now, not everyone can afford that. Only people who can afford, they can do it. That means those people who are in the middle class, upper middle class or middle class to above. But those people, what about those people who are below this? And India, many people are below poverty line. What about those people? Because if you see the limitations of food supplements, they're costly. Preparing a healthy and nutritious diet is cheaper compared to using food supplements. And we have side effects like liver damage with excess vitamin intakes. And also these, if you have to buy these food supplements, you need medical prescription. And mixing certain vitamins and minerals may cause interference with absorption. Like uh, generally, ladies are advised to take iron capsules and also calcium. Calcium because the bone density starts reducing, and calcium, iron because they're enemy. Now, if you take the combination of iron and calcium together, it will not be absorbed by the blood. So, the doctors always advise that they have to take iron at one time and then calcium at other time, and those two should not be combined, be taken together. But many people do not know about this. So, though they are taking that, they will not be able to get it. Then, food supplements are less healthy than food as they provide only nutrient and they cannot provide good fiber as you consuming whole grain or liquids. So, it is just giving you that particular nutrient. It does not help you in your digestion or uh, proper movement of bowels. So, if you see the scenario of food fortification, different countries have started for food fortification in different formats, in different forms, through different channels. Worldwide, all of you know that we have iodized stuff. That is again food fortification, classic example. In Zambia, we have sugar which is fortified with vitamin A. In Bangladesh, we have vegetable oil fortified with vitamin A. Nowadays, we are getting even in India. In India, wheat flour is fortified with iron and folic acid. In African countries also, wheat flour is fortified with iron, zinc, and folic acid. Pakistan, like India, similar condition, iron and folic acid in wheat flour. In China, iron is fortified in soy sauce. Then micronutrient powder, it is available as powder form, which is a combination of all these. So those tablets which we take multivitamin, mineral tablets, these are nothing but these are the things. Now, if you see how these micronutrients can be used, uh, different kinds of micronutrient interventions are available, like they are used through supplementation, or through fortification, or through dietary diversification, or through infant feeding, that is nutrition education, public health measures, all this. Supplementation means medical, by the prescription of the doctor, there you are trying to get the supplements. Or dietary, how dietary diversification. From that means you are trying to eat different kinds of food so that you get different minerals or different nutrients in different quantities. Now fortification, you may be doing at your home level. Then industrial fortification, that just now we have seen like iodized salt and all those stuff. Fertilization, even this is also seen in case of fertilizers when they apply zinc sulfate, ferrous sulfate, they spray that on the crop and see that it is going to give you high yields, etc. Now, this today we are trying to speak about this biofortification. We have learned something about this. Dietary diversification has said that micronutrient rich crops and micronutrient availability in meals. So, if you, before going into biofortification, if you see what is the difference between fortification and biofortification, fortification it is the practice of deliberately increasing the content of an essential micronutrient, maybe vitamins or minerals, including trace elements in a food, so as to improve the nutritional quality of the food supply and providing public health benefit with minimal risk to the health. So that is what we do in fortification. Now, biofortification, it is the process by which the nutritional quality of food crops is improved through either agronomic practices or conventional plant breeding or through modern biotechnology. So, if you see biofortification, it differs from conventional fortification in that it aims to increase the nutrient levels in crops during their plant growth rather than through manual means. We are not adding from outside. We are making the plant to accumulate that particular mineral, whatever we are intending to have, in larger quantities during its growth stage. So, biofortification may therefore present a way to reach populations where supplementation and conventional fortification activities may be difficult to implement or limited. So, this will help in reaching the masses. Just now, I spoke few minutes before that there are many people who are below poverty line. How do they get this? They cannot go and get the iron tablets or calcium tablets or vitamin A pills, etc. So they have to get these nutrients through the food they consume. So biofortification is the way which can address the malnutrition through the staple foods. Behind for it. Okay. So if you see the meaning of biofortification, it is a Greek word bios means life, and the Latin word fortificare means make you strong. So this biofortification is defined as breeding of food crops that are rich in bioavailable micronutrients. So the process of increasing the bioavailable concentrations of an element in edible portions of croplands through agronomic interventions or genetic selections is nothing but the biofortification. And from where this concept of biofortification has come. So I just wanted to take you along to know about the history of biofortification. An economist at the IFPRI, that is International Food Policy Research Institute, by name Howard Boyce, he he was doing some research on he is doing the diets of poor households and nutritional outcomes in Asia. Particularly, you want to see how nutrient intakes were influenced by food prices and household income. So he just was trying to study what are the factors affecting these things. And in general, economists believe that the number of calories was the primary dietary factor. That means if we eat more, you get more diet, more number of calories. And this is constraining better nutritional outcomes in developing countries. This was the underlying premise of the Green Revolution. But in late 1960s, Green Revolution allowed farmers to increase their agricultural output, mainly cereal grains like rice, maize, wheat, etc. Then Dr. Boy's research showed that the differences in diets between the rich and the poor were explained overwhelmingly by the level of non-staple food consumption. Just now I said Green revolution has increased the production and productivity in the staple crops like rice, wheat, maize, sorghum, pearl millet, all these things. But the differences in the diets between the rich and poor are reflected not in the quantity of the staple foods they are taking, but in the quantity of the non-staple foods they are taking. So that is what is determining the health. Moreover, the extra vitamins and minerals in these non-staple foods and animal products were more highly correlated than calorie intake with health outcomes. So, both rich and poor, they are eating carbohydrates through the staple foods. But what the rich are taking extra, they are taking vitamins and minerals through maybe fruits, different kinds of vegetables, and different things, which the poor is not able to take. So, Dr. Boyce concluded that mineral and vitamin deficiencies, not the calories, were the main constraints to better nutrition and therefore to healthy and economically productive lives. So, this study has resulted in this conclusion that not the calories, minerals and vitamins are very, very essential for health. So, in other words, what if we could produce a new variety of seed that was higher in not only your carbohydrates but minerals like iron or zinc. That is how this concept of biofortification that because this the results of this study has made people think how to help the poor not only with the calories but also with the other things. Therefore, farmers and consumers would not see or taste the extra iron or zinc, but their intakes would be substantially increased along with the staple crops. In addition, smallholder farmers growing the more nutritious staple crop would be consuming it as a then they are going to be benefited. Not only humans or animals need these elements, even plants need these minerals for their nutrition. And research has shown that wheat seeds with higher gene content are more vigorous and viable. Not only in wheat, many experiments in different crops have shown that these minerals are essential for even plant growth. Now, the intersection between nutrition and agriculture suggested that if we could find a way to increase the vitamin and mineral content of staple foods grown and consumed by smallholder farmers, we could solve the problem of hidden hunger. And that intersection between nutrition and agriculture has led to biofortification. So, this is the genesis of biofortification. Before going to actually what is happening, if you just go through the advantages, it is biofortification is built on what poor households grow and eat. That is, it is focusing on staple foods. The one-time investment to develop seeds that fortify themselves keep recurrent costs low, and the germplasm of the living tissue from which plants can be grown can be shared globally, making it highly cost-effective. It is self-explanatory. Otherwise, if you're producing, it has to be transported to some place, and then the process has to be repeated to produce in large quantities. But here we're trying to develop those seeds which can accumulate, synthesize more of these nutrients and help. And the recurring costs and the maintenance costs are very less. And biofortification is sustainable. Long after people stop thinking about biofortification, farmers continue to grow and eat their biofortified crops. Now thought of as just only crops, but they are biofortified crops. Biofortification reaches the country's most vulnerable people living in remote rural areas with no access or money for commercially marketed fortified foods. And biofortification produces higher yields in an environmentally friendly ways. And this biofortification is the one solution to reducing hidden hunger worldwide. And it is the process by which the nutritional quality of food crops is improved through agronomic practices or conventional plant breeding or modern biotechnology. Now, if you see the science of biofortification, we all know that conventional plant breeding began some centuries ago, quite long back. Early farmers chose the best looking plants and seeds and saved them for the next year's plant. As the science of genetics was better understood, plant breeders were able to select certain desirable traits in a plant to create improved varieties of plants. The projects and biofortification around the world focus on three crucial micronutrients that are most limited in the diets of the poor. And those three crucial macronutrients are vitamin A, zinc, and iron. So, efforts have been initiated to incorporate these three in their breeding programs so that the plants or the varieties they develop in future would have these things and these nutrients can be accumulated in their tissues as they grow. So the first step in developing a biofortified crop is screen the existing crop varieties and accessions in the global germplasm collections, then evaluate the genetic diversity and identify the plants with the desirable characters. And for this, the first important thing is the source of genetic variation is important for the next step in breeding. So all the plant breeding students know that creation of variation and selection of variation, these are the two basic principles in plant breeding. The same principles of plant breeding are applied here. But there we are looking for yield or resistance or something else. But here we are looking for variation in the nutrient level. Suppose we are looking for iron, we have to see and we have to screen the available germplasm for iron content and then identify those materials with high iron content. Pre-breeding is required if macronutrient variation is discovered in the strategic breeding. Suppose if you find that iron content or zinc content are high in some related species or related genera, maybe your secondary gene pool or tertiary gene pool. Tertiary gene pool is very tough. Even in primary gene pool also, varieties okay you can cross, but if it is in secondary gene pool, you need to incorporate that particular trait through pre-breeding. If micronutrient variation is present in the gene pool, materials may be used directly to develop new varieties. Then parental lines with high vitamin or mineral levels are crossed several generations to produce plants with desired micronutrient content and other traits important for acceptance by farmers and consumers. And then once we have that material available with us, next step is to thoroughly test it. The newly selected micronutrient dense lines in experimental stations and farmers' fields. Then the plants are evaluated for their yield and other agronomic traits, including tolerance to pests and diseases. Otherwise, if I produce a variety with a high iron or zinc level, but it cannot produce high yield, it is susceptible to diseases, what is the fun? So, it should also have the agronomical superiority like what we see in other varietal improvement programs, along with resistance to diseases and pests. These evaluations are done in comparison to the best available local cultivars, like we have the checks. Here also we have to use the local checks and the national checks, whatever, wherever we have those checks available. Influence of varying climatic and soil conditions is also studied, as well as performance and the different agronomic management practices, like fertilizer application, irrigation, etc. All this, just like the varietal evaluation we take up in the coordinated trials, the similar kind of testing has to be done once we come out with the biofortified material. The seeds of the best performing materials are then multiplied and made available as public goods to the national governments, which officially release the new crop varieties and make them available to the farmers. So this is how it is being done. So when we are trying to breed for better nutrition, some points which we need to consider, like aim at combining high nutrient density with high yields and high profitability. So it is not just it has high nutrient content, just go blindly and develop some variety. It does not uh, give you any meaningful variety because without yield, the farmer is not going to accept anything. Then the micronutrient enrichment levels. So if you think that that particular variety has iron or zinc content, whatever target nutrient you're trying to, it should have higher quantity. It is not just an increment level or a minimal increase. And then stability of the micronutrient enrichment traits. So if you find that a particular variety has high iron, it should be stably expressed. That means across locations or across years. The multi-location testing over years and over locations has to be done. Then only we'll be able to know the stability. Then the efficacy also must be demonstrated that it is available. And then adaptability of biofortified crop. Okay, as a breeder, as a researcher, you might have come out with a good variety, but it has to be accepted. How it will be accepted? It has to be accepted by the farmers. They have to grow it. They should be satisfied. Yes, it is giving good yield. Then it has to be accepted by the consumer because it has to be marketed, and then the consumer who buys it also should be able to relish it and feel yes, it is something good. Then only the adaptability of the things come. So ways to breed for micronutrients. The plant breeding of crops with an optimum concentration of micronutrients in the edible portion. As a plant breeder, we'll think, okay, is that mere breeding for higher micronutrient concentration in the edible part is enough? That's not how to develop a biofortified variety a breeder will think of. Then the nutritionist thing comes into picture. No, dear plant breeder, it is not. Then he'll ask why. And they say bioavailability also matters. Suppose I develop a variety with some 100 ppm of iron, but if it is not bioavailable, then what is the fun? Bioavailable means it has to be absorbed, it has to be taken up by the system, the human system. Then only it is useful, otherwise there is no fun. Then breeding for low anti-nutrition factors like trypsin inhibitor, then glyphosate in brassica, trypsins in soybean. Then breeding for nutrition promoting factors. These are all the things which one a breeder has to look into. Then the breeding approaches for biofortification, you can go either through conventional approaches or the biotechnical approaches. Like biotechnological approaches, maybe the marker resistant breeding or the transgenic breeding. So whatever I have told you just now, it is represented here. So you screen the material, genetic diversity for micronutrients. If you are going through biotechnological pathway, maybe you'll go for QTL analysis, end up with gene discovery, or end up with trait specific molecular markers. If you identify the gene, then you will also try to, in later studies, you can go for allele discoveries. Once you have genes and alleles, through genomic analysis, from the diverse material, you can go take the help of bioinformatics and functional analysis. Again, come out with genes and alleles, and then through either genetic engineering, you can go for development of improved cultivars. So this is how the biotechnological applications can be used in for developing biofortified crops. So the challenge in biofortification is putting together all of the pieces of the puzzle to create a micronutrient-rich crop that is acceptable to both farmers and consumers. So first it has to be discovered. That means the source has to be discovered. Then the variety has to be developed. Then it has to be disseminated. So it is just not you develop a variety, but you have to see that it has to be disseminated and it reaches the target. The target is here the consumer or the poor person. So there are different biofortification projects taken up in different crops. Iron biofortification is done in rice, beans, sweet potato, millets, cassava, and legumes. Zinc biofortification is done in wheat, rice, beans, sweet potato, and maize. Provitamin A, that is carotenoid biofortification, is done in sweet potato, maize, and cassava. Amino acid and protein biofortification is taken up in sorghum and cassava. And in India, we have biofortification projects funded by DBT, ICR's project, CRP and biofortification, and also Harvest Plus project, which is done in different universities through ICRISAT, IRRI, then CIMIT, many other city institutes. In India's ICRCLP biofortification, the different crops included are rice, wheat, maize, pearl millet, small millet, sorghum, all these crops. And these are the crops which are being taken up by harvesters. Iron beans, vitamin A maize, zinc maize, iron pearl millet, zinc wheat, vitamin A cassava, vitamin A sweet potato, and zinc rice. So today, biofortified crops including vitamin A, orange sweet potato, iron beans, iron pearl millet, vitamin yellow cassava, vitamin A, orange maize, zinc rice, and zinc wheat have been released in more than 30 countries and are being tested and grown in more than 40 countries. So a significant progress has happened. Crop improvement continues to develop varieties with higher levels of vitamins and minerals that are adapted to a wide range of agroecological conditions and ensure that the best germplasm for climate adaptive and consumer preferred traits continue to be used in breeding biofortified crops. As I told even in the previous two or three previous slides, the challenge in biofortification is putting together all these pieces of the puzzle to create a micronutrient rich crop that is acceptable to both farmers and consumers. But it is just not that the breeder has a fancy to develop a biofortified variety, he will develop and forget. No, it has to be accepted both by farmers and consumers. Now, different uh, countries have come up with different base levels for different nutrients, and they have fixed this is the crop, this is the nutrient, these are the baseline levels, and these are the levels achieved. So in rice, the for zinc, the baseline level they wanted is to have 12 to 16 ppm, and they achieved more than 20 ppm. Protein 78, more than 10. Wheat, iron, zinc, and protein, these are the different levels. Maize, lysine, tryptophan, provitamin A, these are the different levels they fixed. They wanted to have the baseline, but these are the levels as well. In pearl millet, iron and zinc, 45 to 50 ppm, and then zinc 30 to 35 ppm. But we have material which is having more than 40 and 70 respectively in zinc and iron. In lentil, zinc and iron. In vegetables like cauliflower, beta carotene, it was very negligible, but now they have come up to more than 8 ppm. In sweet potato, these are the nutrients, these are the levels. Pomegranate, vitamin C, iron, and zinc. And then I also mentioned about that anti-nutritional factors. In mustard, it is glucosinolates. Previously it was more. They wanted to bring it to less than 120 ppm, but now it has come to less than 30 ppm, very significant decrease. Erucic acid from more than 40% to they brought it down to less than 2%. In soybeans, Cunard trypsin inhibitor, they wanted to bring from 30 to 45 mg per gram of seed meal to negligible. Now they have brought it to negligible quantity. So I will just take out some examples in different crops. So in rice, these are the nutrients, the target countries, the lead institutions, year of release. So for zinc, Bangladesh, Brazil, provitamin A, Philippines, Bangladesh, iron, Bangladesh, etc. So different lead institutions in which year they have been released, all these things are available in these slides. Now, the classic example in case of rice, everyone using transgenic approach, how the biofortified crops are developed. Golden rice is one of the classic examples. It is one of the solutions for vitamin A deficiency. So, what is golden rice? It is a genetically engineered rice which can produce and accumulate beta carotene. That is nothing but provitamin A inside the rice endosperm. If you see the rice, then this is the structure. White rice is like this, but because of the accumulation of beta carotene in the rice endosperm, the golden rice will be looking like this. So the science behind golden rice, the particular genes have been taken: phytoin synthase, lycopene cyclase, and phytoin desaturase. These two genes are coming from daffodils, and phytoin cyclase has come from Erwinia bacteria. And these three genes have been inserted into different plasmids, and they were transferred into agrobacteria, and by the transformation using that particular transformed agrobacteria. The rice plants have been again transformed, and then they got different versions of golden rice, and the first version of golden rice was there like that. Later on, we have different versions, and you all know that golden rice was developed by Professor Ingo Quadratus. Then other scientists along with him, Dr. Peter Bayer, and there are other European scientists who were involved in development of this golden rice. And I'm not going into detail of all these things, but I think many of the students would be knowing about this. And after that, we have golden rice 2 version, where the quantity of the vitamin A, this beta carotene, is in enhanced quantity. Then, if you see versions of golden rice, the first prototype, the beta carotene was only 1.6 micrograms per gram. In the golden rice one, it has 5 to 7 micrograms per gram of beta carotene. Now in golden rice 2, they have replaced the daffodil TYS gene with maize gene. It has resulted in 31 micrograms per gram. So improvements are going on. So you have, this is the wild type, the first golden rice version 1, golden rice version 2. You can see the difference in the intensity of that particular yellow color, golden color. So I just now mentioned what are the differences between golden and 1 and 2, the same. In India, we have also from CRP biofortification program, many varieties have come. Like rice CR Dan 310, this is a pure line variety. It is having 10.3% of protein, very high content of protein in it. And this was developed by National Rice Research Institute, Cuttack. Then there is rice DRR Dan 45. This is also a pure line variety. It is having 22.6 ppm of zinc, and this was developed by Indian Institute of Rice Research, Hyderabad. Coming to wheat, the target traits are zinc and iron. Target countries, these are the countries which try to develop biofortified wheats, and these are the institutions which help them, and these are the different years of producers. The targeted level here was 37 ppm, but China has come up with 44 ppm of zinc concentration. There are different biofortified wheat varieties which were developed at BHU along with collaboration with Harvest Plus and Summit. Then ICR also has got wheat varieties. Wheat WB02, it is rich in zinc. Hello? Hello? Uh, okay. हेलो से मतलब कहाँ ओके ओके
Or
because of some technical glitch I had to stop in between. We were discussing, we were talking about the wheat varieties. Can you mute the background voices? Okay. WB02 is a wheat variety which has iron 40 ppm and zinc 42 ppm. It was developed by Indian Institute of Wheat and Barley Research. Then HPW01, this is also a pure line variety with zinc 40.6 ppm and iron 40 ppm. It was developed by Punjab Agriculture University under ICR ESRT on wheat and barley. Coming to maize, provitamin A was the target nutrient. different countries, Zambia, Nigeria, Brazil, and these are the lead institutions, and these are the years of release in the international level. The target level was 15 ppm, and then the varieties they produced more, 25% of this thing, and the variety was found more efficient, 3 to 6.5 to 1 as against 12 is to 1. So this is also significant development in maize. In India, we have Pusa Vivek QPM9. This is a hybrid. Provitamin A 8.15 ppm, lysine 2.67, and tryptophan 0.74%. This was developed by IARI. Then maize Pusa HM4 improved. Again, this is also a marker assisted developed hybrid. Uh, uh, Here, tryptophan is 0.91% and lysine is 3.62%. Again, this was developed by IARI. Then Pusa HM8 improved with increased content of tryptophan and lysine, again developed by IARA. Pusa HM9 improved. This was also developed by IARA with tryptophan 0.68 and lysine 2.97%. Then sweet potato, the target nutrient is provitamin A. These are the different countries and these are the lead institutions which helped in, and they have come out with the material in these years. So you can see the difference. This is the normal sweet potato, and this is the orange flesh sweet potato, which has enhanced vitamin A content. And you can see the difference in the bread made of this using sweet potato in the breads. The bread is also golden in color. In India, in potato, we have Bhusona, in which beta carotene is 14 mg per 100 grams. This was developed by Central Tuber Crops Research Institute, Kerala, Thiruvananthapuram. And then sweet potato, Bhu Krishna. Again, this was developed by CTCRA Central, that is in Kerala, Thiruvananthapuram. The anthocyanin content is 90 mg per 100 grams. Cassava, provitamin A and provitamin, again here also provitamin E. The target set was 15 ppm, and they could get the bioavailability of beta carotene was about 3.8 to 4.3 to 1, and they could get it to 10 ppm. Then beans rich in iron, iron rich beans. It was done in Rwanda, and the target quantity was 94 ppm. It was released in 2012, and they could get three varieties with 80 ppm. They were released in Brazil. And in different crops, sorghum, the nutrients are provitamin A, zinc, and iron. In coffee, iron, zinc. Banana, provitamin A. Lentil, iron, and zinc. Potato, iron. Just now I have given the examples what happened and in India also. And I also spoke about the anti-nutritional factors. Even reduction in the anti-nutritional factors is also a kind of biofortification. So in mustard, they have Pusa 00 mustard, that even where the erucic acid was brought down to less than 2% and glucosinolates have been brought down to 30 ppm. Similarly, Pusa mustard 30, here also the erucic acid was brought down to less than 2%. Both these varieties were developed by IARA. Then lentil Pusa Ageti Masoor. Here the iron is 65 ppm. This was developed by IRI. Cauliflower Pusa Beta Kesari 1. The beta carotene content has been increased from to 8 to 10 ppm. Again from IRI. Then pomegranate, it is Solapur Lal and the name. Iron is 5.6 to 6.1 mg per 100 grams. Zinc 0.64 to 0.69 mg per 100. Vitamin C 19.4 to 19.8 mg per 100 grams. And this was developed by National Research Center on Pomegranate located in Pune. So this is all about different crops, biofortification in different crops. Now my talk is with relation to iron pearl millet. So now I'll be speaking on pearl millet. If you see pearl millet, it is the warm season cereal crop. India is the largest producer of pearl millet in the world. It has now an area of about 7.5 million hectares, production of 9.73 million tons, productivity 1237 kg per hectare. and it is grown mostly in Rajasthan, Maharashtra, Gujarat, Madhya Pradesh, Maharashtra, all these areas. And these are the different agroclimatic zones of pearl millet, and these are the different places where our ASRP centers are located. If you see, not only pearl millet, millets in general, because uh, we 2018 has been declared as a year of millets, and 2023 is going to be declared as the International Year of Millets. And now everyone is talking about millets. And before going deep into the pearl millet, I would just like to take you along with me about the importance of millets. So millet consumption in India is still day by day increasing because people have accepted and realized that its importance for improving their health, and they are realizing it as a functional food. And if you see health and wellness industry, there is a constant increase. There is a global annual growth rate to 11%, which involves health and wellness industry. So those people who can capture this market are going to be benefited. And not only that, processed food is having a great opportunity. Till now, the processed food items are mostly on rice or wheat or maize. Millets have a great scope to be incorporated in processed food industry. And Indian food market, it is about 1,75,000 crores. So millets can gain into this. Then millets need very less water because they're very climate resilient, hardy, drought resistant, drought tolerant crops. And if you see sugarcane, it needs 2100 liters of water per kg. If you look in the pearl millet, it needs only 350 kgs of liters of water per kg of grain. So the water requirement is very less. And brain drain. The cheapest cereal in India are the most expensive in US. If you see people talk of pearl. In India, pearl millet you may get about 1 kilo for 20 to 25 rupees a kg. But the same pearl millet, if you look in US, 1 pound of pearl millet is being purchased at a price of $4 to $5. Not only pearl millet, different millets, quinoa, all these things are being purchased at a very high premium prices. And if you see the nutritional composition, sorghum, pearl millet, finger millet, they're very rich compared to wheat and rice. And now pearl millet, it is having comparatively very good composition of nutrients compared to other millets, especially when compared to wheat and rice. These are some of the macronutrient contents of pearl millet: carbohydrates, proteins, fat. Even carbohydrates, it has gluten-free starch, then low glycemic index. Proteins are gluten-free, low in lysine. Then fats, 75% are unsaturated. They contain omega-6 linoleic, then omega-9 and omega-3 fatty acids. And proteins of phospholipids are used in brain function. So consumption of these grains is very, very beneficial for your health. And also, if you can see the vitamin profile, you can see a very good vitamin profile in pearl millet compared to wheat, rice, sorghum, and maize. And if you see the structure of the grain, iron is located in the alveolar layer, which is just beneath the pericarp. This is where the iron is present, if you can see here. And the rest of the thing is endosperm, which is having more of starch and then lipids here. Now, as per the source from Harvest Plus, pearl millet daily per capita consumption, if you can see 150 grams per day, then the total conventional pearl millet is getting 47 parts per million of iron. And if you target an increment in iron content, they have targeted 30 ppm. And the biofortified pearl millet, they wanted that it should be minimum of 77 ppm. So the concept of the base minimum increment of iron content as per Harvest Plus, they wanted it to be 77 ppm, to be called as biofortified. And if you see the consumption in pearl millet growing regions, 35% of the total intake of calories, protein, iron, and zinc is coming from pearl millet. And if you see those areas where pearl millet is grown, just now I have shown you in Gujarat, Rajasthan, Maharashtra, Haryana. And if the consumption kg per year mostly consumed, highest consumption is in Rajasthan, followed by Gujarat, then Maharashtra, and then Haryana. Now, as I have mentioned to you that if you have to develop a biofortified variety, we need to screen the available germplasm for those particular target nutrients which we are trying to use in biofortification. So, when we are talking of iron pearl millet, the target rate here is iron. So, how do you screen for iron content? A rapid screening technique was used, that is XRF, and this instrument was there at ICRISAT, and ICRISAT was kind enough to screen all the available germplasm. Initially, they had a Harvest Plus project. Even now, they have a continuing Harvest Plus project. Dr. K. N. Rai was the person who was associated with this Harvest Plus biofortification program in Berlin. And those people have come up with a non-destructive screening technique for screening for iron content. And if you want to screen a large number of germplasm, you should have a cost-effective method. The maintenance cost should be low and it should be quick enough. And if it is a non-destructive type, it is still more better. So they started using this XRF. This is the XRF instrument. And it is very handy and also cheaper. It costed only $2 per sample. It was used only for estimating iron and zinc content. It is safe and simple to operate. That time it was handling only 300 samples. Now they have increased the efficiency. It is relatively cheaper to install and reasonably precise correlation. Compared to ICP and XRF, the correlation is 0.69 to 0.97. So, whenever you want to screen for these nutrients, you should have a standard and stable, reliable phenotyping facility. Here, phenotyping means we are using this particular equipment to estimate the iron content, iron and zinc content. So, as I told you before, they identified the iron rich material, then they made different process, again by cross, they came out with good materials. Initially, then Shakti, a biofortified high iron pearl millet variety was developed in a collaboration between ICRISAT and MTKV Dhule. Then this one was having 71 ppm iron and 40 ppm zinc. And this Dhanshakti is nothing but an improved version of ICTP 8203, which was the variety. Initially, it was released for Maharashtra state in Zone, but later on, looking into its importance because it is a biofortified variety, it has been released to all over India in 2014. Now, this was happening parallelly, and ICR also had started a program, CRP and biofortification, with two objectives. With the objectives for pearl millet development of pearl millet genotypes for high zinc and iron through conventional breeding, and identification of genomic regions associated with enhanced micronutrients through mapping and association mapping. Then development of value-added products for ready-to-cook and multigrain mixtures suitable for midday meal scheme. So these were the objectives for pearl millet biofortification under CRP biofortification program by ICR. There were 6 centers in this particular program. These are the 6 centers: ASRP pearl millet PC unit at Jodhpur, Jamnagar, Hisar, Rahuri, IR in New Delhi, and Durgapura. And these are the people involved in this program. And under This category of biofortified pearl millet, we have 6 hybrids which have been developed. The first one is Dhanshakti. Just now I have shown you that it is a variety. And these are the other 6 hybrids: HHB 299, AHB 1200, AHB 1269, HHB 311, RHB 233, and RHB 234. These are the different biofortified pearl millet hybrids developed by different research centers under All India Coordinated Research Program on Pearl Millet. And you can see the iron and zinc contents. In HHB 299, it is 73 ppm iron and 41 ppm zinc. In AHB 1200, it is 77 ppm iron and 39 ppm zinc. AHB 1269, it is 91 ppm iron and 43 ppm zinc. And in HHB 311, it is 83 ppm and 39 ppm. RHB 233, 83 ppm iron, 46 ppm zinc. RHB 234, 84 ppm and 41 ppm. You can see the levels. So this is Dhanshakti, EHB 1200. This is the hybrid, and this was developed by Aurangabad, again in collaboration with ICRISAT and under ICRES in Pearl Millet. So this is the experimental plot of HB 1200. HHB 299. This was developed by Haryana, again in collaboration with ICRISAT under ICRES in Pearl Millet. HHB 311, again from Haryana. This is HB 1269 from Aurangabad. RHB 234 from Durgapura. RHB 233 from again Durgapura, that is Rajasthan. Now, though we have developed biofortified pearl millet hybrids, the yield levels were not on par with the regular checks which we were using. So, what is the fun if a variety or hybrid has been developed with high iron content or zinc content, but it is not having good yield? Also, we do not have a separate market to distinguish or differentiate between high iron pearl millet and a normal pearl millet. If a farmer takes high iron biofortified pearl millet grown in his field and takes to mandi, there are no takers that it has high iron, it will be given a high price. Then what is the fun? He is losing yield and he is not getting a premium price. And we do not have till date in place a market or a structured market economy which can differentiate a biofortified product with a normal product. So in the number one. Number two, the basic purpose of biofortification is to target the poor in the staple food. So if we are trying to fix a premium price for the biofortified product, whether the normal poor farmer will be able to purchase it, and we do not have any well-defined structural system in the market to differentiate. So in the near future, the farmer will not be able to get any premium price for the biofortified product. So keeping all these things in mind, and after some thorough discussions, a decision was taken in 52nd annual group meeting in Ludhiana, where a baseline, a minimum benchmark was fixed that without this much level of iron and zinc content in the entries, whether it is a hybrid or a variety, they will not be promoted to next level. You all know that in All India Coordinated System, we have 3-tier testing. The first year, that is initial varietal trial or initial hybrid trial, where it will be tested all over the locations. And then based on the performance, the high performing or the good performing entries will be taken to advanced level of testing, where they will be tested in those specific ecologies in zones in 2 consecutive years. So you'll be able to get a idea of how it is performing over locations and over years. And the promotion criteria from the first year of testing to second year to third year testing is we have some criteria like we look into the yield that it should be more 5% more than the check or 10% more than the check. Then it should have this particular days to maturity or days to flowering. Then it should have this many, this much of resistance to disease. We have all fixed scales and parameters. Now, anywhere in the world, we did not have or we do not have that uh, criteria, nutritional traits as a criteria for promotion. So that particular first decision of incorporating nutritional parameters in promotion criteria was taken up by Pearl Millet, and we have fixed that iron content should be 42 ppm and zinc content should be 32 ppm. So without this 42 ppm iron and 32 ppm zinc, no variety will be promoted to next level of testing. Irrespective whether it is a very high yielding or a very resistant material, no. Without this much of iron and zinc, they will not be promoted. Beyond this iron and zinc, any much you may have more quantity that will be promoted, but below this, no promotion. And this was appreciated by Harvest Plus, and the certificate of outstanding appreciation was given by Harvest Plus, showing that for the contribution to the National Biofortification Research Partnership for iron pearl millet and for establishing the first global minimum standard for micronutrient in the cultivar release policy that has a significant and long lasting positive impact on the mission of Harvest Plus for improving human nutrition. So this happened 3 years back, and till date, the other crops are still struggling to fix benchmark values in different crops. Not only in India, anywhere in the world, we do not have. Only this is the first case in pearl millet. And this decision was taken in the annual group meeting after a thorough discussion with the public sector breeders and private sector partners. Now, that is the development of biofortified varieties and inclusion of iron and zinc content in the promotion criteria. And because we are talking of pearl millet, I just wanted to take you along what is happening, whether this biofortified pearl millet, which has been bred for high iron, is really addressing that or not. Whether it is beneficial for the targeted consumers or not. So some trials have been taken that when iron deficiency in children, means in children who are deficient in iron, they were fed with biofortified pearl milk in the form of rotis. This lady is giving rotis to the children. After 4 months, we could see a significant difference in accumulated body iron for the high iron biofortified pearl millet group versus the control group. So that means the efforts incurred in developing high biofortified iron-rich pearl millet are really paying returns. The targeted group is getting the benefit. And now, this is one part. Okay, we have developed the varieties. It is affecting and it is good for the targeted community. Now, how to disseminate it? I said developing, then delivery of the product, identify the material, okay, then we develop the product. Now it has to be delivered, it has to be disseminated. So what are the strategic factors driving delivery of iron rich pearl millet? If you look into the national agricultural system, in both public sector and private sector, good strong pearl millet breeding program is there, and commercial market is largely driven by hybrids, and therefore private sector plays a key role. And now the point is both public sector and private sector are acting, the two main actors in mainstreaming iron-rich pearl millet. So to implement that, what are the things we need to look into? Policy reform to ensure that varieties have minimum threshold levels of minerals and vitamins. Number one, we have already fixed 42 ppm iron and 32 ppm. Then establishing economically viable testing procedures for assessing. Already, I have shown you that XRF is a very good facility, cheap, fast, non-destructive. Then policy reformation to ensure farmers to get extra benefits in terms of MSP or premium prices. It has to be implemented because the government has to be convinced that yes, it has to be given, the iron rich pearl millet has to be given premium price. And then policies formation for integration with school feeding programs. If you all know that Millet Mission was launched in 2018, under that the midday meal schemes have to be given to children, including pearl millet and then other millets, sorghum, etc. So in pearl millet also we are trying to push biofortified pearl millet. As we just now I have shown one slide where the children were benefited with iron rich pearl millet. So in the midday meal schemes, efforts are being made to push iron rich pearl millet. And then consider dietary quality and economic policy analysis related to food security. Yes, not just energy intake. The analysis should also say whether it is rich in iron or zinc, and so what is the impact of that nutrient, enhanced nutrient content in that particular variety, how it is impacting the health and economy. All these things have to be looked into. And if you look from private sector perspective, seed commercialization and marketing, these are the two things. The seeds are supplied to the farmers through distributors and retailers. And here the private sector also are pushing their biofortified material in by giving to the farmers in reasonable packs, maybe 1.5 kg or 3 kg packs. And how the farmers are made aware by this, that this particular product is there, it has high iron or something like that, by through demonstrations. The private sector also is doing farmer demonstration. The government is also doing frontline demonstrations, field days, all these things. Then build product acceptance, demand creation of pearl millet grains, flour, value-added products. So now you are growing biofortified crop. If there is no taker for that grain, why the farmer will do? That means we need to develop a chain. How you take that biofortified pearl millet, use that particular grain in developing some good products, food products, then value addition, then selling them. That means a series of people have to be involved, series of stakeholders have to be involved, and they have to be made aware that this particular product has this much of iron and this is beneficial. So everywhere we have to make them aware that this is a enriched product. And now we have some delivery challenges and recommendations incorporating iron pearl millet grains in the existing institutional government programs. Now we say that as scientists we say that this is rich, this is good, this is better, but other party should accept it. So we need to create awareness in the policy makers and also the implementers who are there at the schools to buy these products and then incorporate them in the midday meal schemes. And also developing technologies to increase shelf life of pearl millet flour. All of you know that pearl millet has very poor shelf life. It develops uh, rancidity, that is off flavor and odor after storage. So the pearl millet will not be accepted. It will not be useful for making uh, rotis or any other preparation after 7 to 10 days. So we are trying to develop technologies to address this issue, and already big programs are going on at IAR and Division of Biochemistry, and also ICRISAT is doing with whatever. So the key takeaways are biofortification program should be made integral in all national breeding programs. Already main crops like rice, wheat, maize, pearl millet, sorghum, everyone is doing. The biofortification program has become a main main breeding program. And then the breeding for this trait has to be mainstream. So along with yield, disease resistance, insect resistance, we should also breed for increased iron and zinc content. So mainstreaming of these traits. Then policy reform to ensure that varieties with minimum threshold levels of iron and zinc. We have initiated in pearl millet, other crops have to do that. Then establishing economically viable testing procedures. Already XRF is there. In other crops also people are using them. Then differentiated supply chains for selling the varieties to farmers with branding, labeling and certification efforts. maybe one-to-one or public media or campaigning in whatever way possible they should be done. Organizing consumer awareness and education campaigns and promotion as health foods because they have enhanced nutrient content, so they are health foods. Research and development targeting increased productivity of biofortified pearl millet varieties and hybrids in farmers. Of course, it is being done. This is what I have told what is happening in India. In Harvest Plus also they try to put all these things in a chart form. Whatever I told now, the same things have been reflected here in different terms. This is one publication where portion. This publication was brought out by Division of Biochemistry at IR and JSRP in Pearl Millet, stating that pearl millet oriented, staple, healthy, all inclusive nutrition. So we just wanted to make people aware that pearl millet is a very nutritious food and people should make use of it. Now, just before I conclude, I just want to bring out 2 3 points. The just now I talked about pearl millet. It is a nutritious food. The particular crop is climate smart. Now, if you see in the COVID-19 pandemic scenario, what we are focusing, we are trying to save the people. That means we are focusing on the survival of the people. But the very survival of a person or an organism depends on the food it takes. That is the nutrition. So when we talk of survival of the people, nutrition is a part of it. And biofortification is shown to improve not only micronutrient status but also the health outcomes, including cognitive and physical function, stronger immune system, and lower the morbidity to a range of diseases. So, the more we take the biofortified foods, the more it is going to help our health or increase our immunity. Now, this coronavirus pandemic has highlighted and it has shown the need to ensure good nutrition. And this COVID is a critical wake up call to rethink, redesign, reprioritize our food systems to deliver nutritious and affordable food to all. And that is possible through biofortification. So policymakers, researchers, and agriculture sector need to come together to systematically solve the food divide that the pandemic has highlighted. Why the people are so many people are succumbing to this? Some people are asymptomatic, some people they're highly susceptible. In the same family, husband is put in ICU, wife is just put under quarantine. Both of them are COVID positive. That means there is a difference in the immunity. It depends on the type of food you take. Maybe their wife and husband only few years back, but right from the development, from the childhood, the kind of food those two people are given, that shapes their immunity. That shapes them what they are. So, good food with good nutrition is very, very important. So, thanks for your attention.
Thank you so much, ma'am. It was very effective talk. Now I will take some of the questions. It will come in
Okay.
your screen. Ma'am, can you see that? Is the
No,
biofertilizer crop consisting all types of? Ma'am, it will be in your screen visible.
I'm not able to see. Should I close my presentation then?
No, no, no.
Okay, okay. Now I'm able to see the same in my yeah.
Yes,
Is it
in the,
biofuel consists all types of essential nutrients which fulfill human body requirements? Okay. See, all crops, biofortified crops, it depends on the type of nutrient which we are targeting. I mentioned that three nutrients, iron, zinc, and vitamin A. These are the three major nutrients which are affecting the health. So at present, people are targeting only for these three. But if someone is interested in developing some other crop with some other desired nutrient, they can go. But at present, efforts are only towards this. I mentioned I also showed you in some cases with high protein content, reduced anti-nutritional factors. But all crops will not be addressed, all nutrients will not be addressed by biofortification. We need to take a balanced quantity. Next one,
Then that is
which,
which is the best. Yeah.
uh, which is the best approach for biofortification of crops? Conventional method is the best method, but to if you do not have any means sources of enhanced nutrients in the germplasm, then you may have to create variation, maybe using molecular technology or transgenic technology. Like the example of golden rice, there they have used the transgenic technology. In case of maize hybrids, I showed Pusa hybrids have been developed. They have taken the help of market technology. So depends. If you have the sources with enhanced nutrients, the conventional method of improvement is the best method. Then what is the adoption rate of biofortified varieties among farmers? Okay, I mentioned in the initial slides that a farmer when he grows he will also consume. So now only few people have started growing across the world. I told you that 30 countries have started growing and 40 different crops. That is how I told. So slowly it is increasing. The farmers are also consuming and it is going to the market and then the consumers are also going to benefit. Next question is, by doing biofortification, yield parameters get retarded to a great extent. So how can we combat with this problem? That is why I told you that when we start breeding for biofortification, as a creation of diversity. If diversity is there, variation for the nutrient content is there, then we have to use it in development of a variety. After developing a variety or a hybrid, it is not that we should just go and release it as a variety. I said that it has to be compared with the checks for its performance and also resistance to diseases and insects. So then only it will be taken. Otherwise, there is no point in just merely developing a fortified variety. It has to be released as any other variety is being released in the ESRP testing. The next question is, you mentioned about iron biofortification. We know iron is more sensitive to environmental variations. How to cope up with such problems? So yes, iron is sensitive to environmental variation. We do find large amount of variation in the data coming from different centers. So what we do is we test that material in different locations for years, and then we take an average. In different locations, maybe in Gwalior it may be giving 80 ppm. The same variety may be giving 60 ppm in other locations. So I will not just go by the data of Gwalior. So I will take the data across locations and across years. Then only we will go further. As compared to common crops, is biofortified crops cost effective? Yes. If you are going by conventional methods, they are going to be cost effective. Only investment is for the screening, initial screening. Otherwise, later on, we don't need any other special technique. Just like any other crop, like any other variety, they'll be propagated. So they are definitely going to be cost effective in the long run. Any others?
Ma'am, a lot of questions are there. Actually, I'm picking some of them. Na? But last question, ma'am.
Is breeding of nutrient deficient crop the right solution to global issue of mineral malnutrition? Why not? Yes, that is why it has been started. All people cannot afford to go for buying fortified foods or take up supplements. In my initial slides, and I mentioned about that the IFPRI study also, there is a divide between the rich and poor because the rich were able to take up non-staple foods which are going to give them minerals and vitamins, and the poor were not able to get because they are not able to afford to buy those fruits or other things. Similarly, they will not be able to afford to buy the supplements in the market. So if we can incorporate these essential nutrients in the staple food crops, they are definitely going to be a address this issue of mineral malnutrition. Definitely, that is why this particular biofortification thing has been started.
Thank you, madam. You have given a lot of information about biofortifications and a lot of deliberations also. Now, very much on the behalf of RVSA, I once again thank you to you, sir, madam. Okay.
Thank you.
Yeah, of course.
Next, so our next speaker and the last one, Dr. P.M. Gaur. He is Director, Asia Research Program, EPSET, and he will give the lectures on through this webinar, Advances in Chickpea Improvement. So, Dr. P.M. Gaur is the Research Program Director for EPSET, Asia Program Hyderabad. He is basically a plant breeder and geneticist by training and has over 3 decades of experience in chickpea genetics and breeding. He did PhD in crop science from the University of Saskatchewan, Canada, where he was a Commonwealth scholarship and developed the first genetic map of chickpea. Before joining ICRISAT, he was professor and principal scientist of chickpea breeding at JNKV Jabalpur. He initiated chickpea breeding, particularly Kabli type, at JNKV Jabalpur and developed two varieties very popular, that is JGK1 and JGK2, which goes to his credit. He induced and identified several mutants in chickpea and was first to report genetics of many traits. Dr. Gaul led existed chickpea breeding program for 17 years. During this, he developed 65 chickpea varieties were released in 8 different countries from explicit bred material. Out of them, 25 varieties in India. He is an adjunct professor at University of Western Australia and to this day RVSK University Gwalior in India. He is recognized supervisor of students of 14 universities in 5 countries. He has several credits to his award. He received Indian Society of Pulse Research and Development Recognition Award in 2007, Honorary Fellowship in 2020, Outstanding Scientist Award for Research in 2010, Jawahar Ratna Award from JNKV Jabalpur in 2018, Doreen Margaret National Award from ICRISAT 2019. He has more than 170 articles in peer-reviewed national and international journals and edited 2 books and so many chapters in proceeding and books. So I request Dr. Gaur sir to kindly start your lecture to benefit us. Okay. Thank you.
Thanks, Dr. A.K. Singh. It's really a pleasure to give this presentation, and I'm really thankful to the university, specifically to Dr. Rao, the Vice Chancellor of the university, Dr. Manoj Tripathi, and Dr. Sushma Tiwari for organizing this. So, in this, let me see if are you able to see it?
Yes, sir.
Huh?
Audible.
Okay. Can you see my presentation? Because I am not able to see others now.
No, 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 sir, it's not visible.
Sir, on your presentation, please once click it and then share screen will come. Your
Yeah.
mail is being seen. We are seeing your mail. Please come out, close it.
उनको प्रेजेंटेशन करना है
Sir, your email is open.
Oh,
यही तो मेल खुला है हमारा तो
can you see now?
सुना
It shows here that I am sharing.
Sir, but you need to once click on that small screen and then it will come down share.
I think we should
Someone else is also sharing their screen. Please come out of it.
Yes, sir.
Yes, sir. Oh, yes.
Okay, I'll try again.
Please, sir, share the screen, sir.
Yeah, I'm just doing that only, and I'm sorry.
Once click on that screen, sir, anywhere, and then click share.
Sir, sir,
Yeah,
please click on share screen option located at bottom of the screen where the mute
yeah.
stop
No,
frame cam
no, I did
option.
that.
So
Yeah,
you click on share screen, sir.
yeah. Then I
Uh,
said share screen.
then one dialog box is displayed on your screen.
Ah,
That share button also located. So click on that dialog box, then share button.
yeah. Can you see now? Are you
Yes,
able
sir.
to see now?
Yes, sir.
Huh?
I'm giving you permission, sir. Please. Okay, sir.
Okay. Thanks very much, sir. Sorry about those technical difficulties. But when I am sharing this screen, I can't see anybody. That's fine, I think.
but we can see you sir
Okay, fine. That's fine. That's fine. I'm sorry about that. We lost some time in that. So the whole idea of this presentation was to give an overview of the advances that have taken place in chickpea. So what I have done in this presentation is, organized in these. This is the outline. So there has been a significant change in the scenario of chickpea cultivation in India. I would like to talk about that because chickpea improvement priorities, directly depend on those changes. And then briefly touch on advances that have taken place in chickpea improvement and what can we do to further improve efficiency of chickpea breeding program and a little bit summary of that. So if you see chickpea production scenario, and we all know we contribute about 2/3 to production and area both in chickpea. But in this pie chart, I particularly want to draw your attention that in recent years there are countries which have come up here which were not earlier there, like USA, Ethiopia, Myanmar. These countries, their production was not very high. Just in federations, these are the new countries where there has been a lot of increase in production of chickpea in recent years. But still, we continue to contribute 2/3 of the production, and Australia continue to remain second largest chickpea producing country. Now, if you see the last 10 years, the growth that has taken place in chickpea production, and because we contribute 2/3 of the production, the global trend is just just mirror image of our production. And there has been, you can see that India, we had, 58% growth in area and 98%, almost double the production in last 10 years. And that's how the world is. But here I want to emphasize that not only India, but if you take the rest of the countries which contribute 1/3 of the production, they also made good progress in recent years in chickpea production. Now, this is where what we have discussed many times. There has been a significant change in the scenario of chickpea production in India. Earlier, if you see the north state and northeast states, they used to have larger area, about 4.2 million hectares. But because of the expansion of irrigations and wheat and other high value crop, this area continually declined. The chickpea was replaced by wheat and other crops. So this area now has only 0.7 million hectare. But this is amazing, amazing. You can see in central and southern India, our production from 2.2 million hectare area to from 2.2 million hectares increased to 7.8 million hectare. You can just imagine more than 5 million hectare area was increased, in these central and southern India. So a big change in chickpea cultivation in India now. And another point I want to emphasize is that if you see the total production figures of India over the years, you will see that only in recent years we made a good progress. But this happened because we were compensating for the loss that was taking place in northern India in area. But if you remove northern India and if you take central and southern India, and if you see the graph, from beginning we were making very good progress actually in chickpea. And if you compare it with the rest of the world, if you remove India and see the rest of the world, in fact, we were doing better than the rest of the world in terms of chickpea production from very beginning. And if you see over the years from 60s to till now, in India we have 220% increase in area if you take only central and southern India, where the world has only 96%. And see the production, 577% increase in production, whereas the other rest of the world 235% increase in production. And yield also, we made good progress, almost double the yield. Here 71% increase in the yield. So a point I want to make is that when we see the total picture, total area and production of country, we don't see that, because the loss that was taking place in northern India and northeast India for chickpea area, we were compensating that. But if you remove that, you can see that India has done remarkable work in chickpea production. And this is the current scenario now. We have Madhya Pradesh is contributing 41 from beginning. Madhya Pradesh has been the largest contributor. And then, you can see other states coming up, Maharashtra, Rajasthan, Karnataka, Andhra Pradesh. You can see these south states, and see now Uttar Pradesh, 5% contribution, which is now, AP is contributing as much as the UP is contributing now. So this has been a change in scenario, significant change in scenario of chickpea production. Another change that had taken place is over the years is the increase in the range of sowing dates. This was amazing to see that even in southern India. This picture I took long back in Andhra Pradesh. We were driving there and we just saw this farmer's field. We don't know even that these three fields are same farmers or they're different farmers, because the farmers were not there. We just took the picture, and amazing to see that even in Andhra Pradesh with short duration environment, you have a crop, chickpea crop ready to harvest. You have a crop which is close to maturity, and then another crop which is just at seedling stage. So you can see the kind of variations you have in sowing date in chickpea. Another is that the possibilities or our efforts in expanding chickpea cultivation on rice fallows. So we have to take into account the crop breeding program or crop improvement program, chickpea programs have to take into account these, requirement which have been changing, and the change in area, expansion of chickpea in central and southern India. Also, there is another factor which is important, many times ignored, is that, of course, we have been importing chickpea. We have been always largest importer, but you can see in recent years, we have started exporting chickpea. And you can see this is the average of 5 years, 2013 to 17. We were the third largest exporter of chickpea. And this is happening because we have increased our Kabuli chickpea production, and there is international demand, and our local demand is less, but our production is high, and we have opportunity of exporting Kabuli chickpea to another country. So this is also we have to take into account in the breeding program. Now, coming to the priorities, of course, enhanced yield potential always remain the number one priority, but the others are very important. Early maturity in case of chickpea, because the chickpea is expanding to short season environment. Then these are the, abiotic and biotic stresses. If you take abiotic, main is, drought, and then heat stresses. And in diseases, you have fusarium wilt, ascochyta blight, and the pod borer. Then in recent years, emphasis on suitability to machine harvesting and herbicide tolerance, market preferred traits, and higher protein and micronutrient contents. So these are the priorities for crop improvement program. And I will just briefly touch on some of these. There is a, in chickpea, significant progress has been made in development of early maturing varieties. And as I said, that chickpea is largely grown in rain-fed conditions in post-rainy season. So normally the crop, experiences drought at the reproductive stage and terminal end. And as I said that, because of the sowing date fluctuations, chickpea moving to short season environment of southern India and in rice fallows and climate change, you get also, the chickpea is also exposed to high temperature during reproductive stage. So heat stress is also becoming very important. So there has been, I will say that very good progress in development of early maturing varieties, because they help in escaping these end of the season, stresses, drought, terminal drought, and reproductive stage heat stresses. So even in early, you have the categories, you have early, you have extra early, even you have super early. But you have to select based on the, the growing length of the growing season, favorable growing season. It's not that you always need, extra early and super early. You have to match your phenology of the crop with the available favorable growing period. So, and generally it is said that, in the crops, if you reduce the duration, you have less growth and you have less, the productivity less. So there is a penalty on yield. This is the general, case in most cases. But because chickpea is indeterminate, has an indeterminate growth habit, it has a plasticity. So if, there is a favorable growing environment, means there is a moisture in the soil, then it expands. So the plants will continue to, grow, flower, and pod. So that's the advantage in chickpea because of indeterminate growth habit. And because that, it has been possible to develop high-yielding varieties, and early maturing. And there are many examples you have, and the the Madhya Pradesh RBS KVV and Jain KVV, they have played great significant role in developing these varieties and actually enhancing production and productivity in central and southern India. One such variety which brought revolution, developed by RBS KVV earlier with Jain KVV, is JG11, which really brought revolution in Andhra Pradesh, replaced the 40 years old variety Annagiri completely from Andhra Pradesh, also spreading in Karnataka. Now, there's a lot of work done in terms of understanding genetics of flowering in chickpea. And by classical, allelic test, we were able to identify 4 different genes for earliness in flowering. Means you can use any gene, will have similar effect. And you can see that, how you can do, this is super early, another super early, you cross, and you got a very late F1, showing that they have different, genes for earliness. So by this simple test, we were able to identify that 4 different genes, EFL1 to EFL4 for earliness in different. And also, our PhD student later on worked on mapping of these genes. And, because, these were major genes, you had, major QTLs which corresponding to those genes in the chromosomes at different locations. Now, the advantage of this is that because they have similar effects, you can see the linkage relationship with other traits, and you can deploy the allele which has, no negative effects with others. I give you an example that, it is generally said that, ascochyta blight susceptibility is linked with earliness. So now in this case, you can find that which gene is linked. So you can deploy another gene, which is on different linkage group. So this is one advantage of using this. Also, in case of wild chickpea, if you take wild species, they are very late in flowering. And when you cross them, they create a challenge because they are very late. You have to match the flowering with your cultivated. So usually what we do is we use vernalization. Vernalization is means you grow, germinate them in petri plates. You put it after germination in refrigerator for a month. That's the vernalization treatment, and then you again transplant them. So you would get early flowering, and you can match very well with your cultivated. So we thought, let's look at the genetics and identify the gene for this vernalization response. It was very clear when we use this RIL populations, which is interspecific cross. There was two, these are two parents and two different peaks you get in the RILs, clearly showing that there is a one gene for vernalization response, and that was successfully mapped on the linkage group 3. Coming to drought tolerance, as I said that chickpeas is still largely grown on residual moisture. Usually, either irrigation is given for sowing, for germinations, and later on no irrigations, and so crop experiences the drought. And it's a complex trait. You have a mechanism for escape, you have a mechanism for avoidance, and you have a mechanism for tolerance. So it's very difficult to identify those individual traits and target them. So generally, breeding programs, they evaluate material under moisture stress conditions, under rainfed conditions, and select for the tolerant lines or higher yielding lines in those environments. At ICRISAT, a lot of work was done to look at the different traits, and it was identified that this genotype ICC 4958, which has the prolific root system, was giving drought tolerance, at least in semi-tropical environment where we have predictable terminal drought at the end. And recently, after that, the same RL populations was studied by our physiologists for shoot vigor traits, and they identified that is the same. It also has the bigger shoot vigor also is in these genotypes. And traditionally, the breeders have used this genotype in breeding program and selected normally for the drought tolerance only by grain yield under drought conditions. And there are varieties released. About 4 varieties have been released by using this drought tolerant parent ICC 4958 in the breeding program. But in recent years, we developed 2 mapping populations. In fact, we started from the root traits because in the beginning we knew that there is big difference in the root systems, and we identified 2 contrasting parents and developed 2 mapping population. And it was interesting to see that you don't have any common parent in these 2, and still you have the same location. You have the QTLs, several QTLs corresponding to that region, which is contributing to root traits and other traits. And then later our physiologists, they further looked at that, and they found that this region is actually also contributing to the shoot vigors, many traits. So root vigors and shoot vigors together, and they say that this is actually QTLs for vigor traits which are there. And this has been very consistent. So we thought we should introgress them in different backgrounds. So we started marker-assisted breeding programs and took mega varieties like JG11, ICCB10, JG16, Jackie 9218. And at IARI, Bharadwaj, I saw him, he is there. And he introgressed in this PUSA 372, this QTL through marker-assisted back crossing. Several of these, lines which were tested in coordinated trials. And of course, here, in this case in India, this RIL introgression lines of PUSA 372 is really performed very well and has been released last year as a PUSA 10216, by Dr. Bharadwaj in ICR IIRI. Similarly, one line JG11, which we supplied to integration line to Ethiopia, our collaborator there, they released one variety from that. So there are two varieties at least released through integration of this hotspot QTL in the different varieties. Coming to heat tolerance, this is one area which is very fascinating, and we never expected that chickpea, which is a cool season crop, will have that big variation for heat tolerance. Normally, the normal temperature for growing chickpea is 10 degrees Celsius to 30 degrees Celsius, and if the temperature increases from 32 degrees Celsius, you start seeing the less spots on the plants. And highly sensitive lines, you will be like this. They will have flowers but no pods if the temperature exceeds 35 degrees Celsius. If you get some cooler days, you may get some pod set, but again there will not be pod. So what we've done at ICRISAT is that we developed a very simple and effective screening technique. So growing another crop, our normal crop we plant in October, but for this heat screening we plant in February. And then when we plant the crop in February, the temperatures at the reproductive stage will be always more than 35 degrees Celsius. So you get very natural screening and very predictable temperatures. So you can see this is in the field screening, and you can see the line. You may say that this is a late, but it is not late line. This produced flower, but didn't produce pod actually. This is a tolerant line. You can see the difference here in tolerant and sensitive lines, highly tolerant and high sensitive lines. So there is a large phenotypic variations for heat tolerance in chickpea. And one of our student from University of Sydney, she worked with us, and she looked at physiological traits. And one trait important is that pollen fertility. You can see this is the tolerant line, and even at 38 degrees Celsius, the pollens are fully fertile. Whereas this insensitive lines, you can see at 35 degrees Celsius, you see the the pollen is sterility. It's actually totally pollen sterile. All pollens are sterile here. And in this case, tolerant, even at 40 degrees Celsius, you can see still you have some pollen viable. So that's the main trait we have seen that affects the heat tolerance. Now there are many varieties which have been released now, heat tolerant, and I want to mention one line which was ICCB 92944. Which line? This is a very good heat tolerant line, and it has been released in 4 countries. In India, it's JG14. In Myanmar, it's JG6. In Bangladesh, it's Vari Chola 10, and in Kenya, it's Chania Desi 2. So this shows that it is showing this heat tolerance in variable environment. Even in I must say that in Myanmar now it is covering 20% of the area. There in India also this is becoming very popular, particularly in the states where chickpea is grown after rice fallows in late sown conditions. And we did experiments in several states, and we found in late sown condition it gives 10 to 25% higher yield than the West varieties. So I believe that now, heat tolerance, we have a lot of good materials. It can be in the mainstream of the breeding program. Means all the varieties we develop, can have now heat tolerance. Our PhD students worked on mapping for heat tolerance, as we had a very contrasting genotypes. You can see here 2 genotypes, heat tolerant and heat sensitive. We developed RIL mapping populations, and the student worked on it. And then we were able to identify 2 major QTL for heat tolerance. We are working on validation of these QTLs, and maybe these will be available for routine use in the breeding program if we want to use. Of course, there are efforts going on for development of genomic selection models in chickpea. So we developed 1 training populations, which was from the elite breeding lines of ICRISAT from international nurseries. We developed them. These have been phenotyped, and Dr. Bharadwaj was involved in it from IRI, and Patan Cheru in rented in irrigated conditions. Genotyped them using 3,000 markers, and the models were 6 models were used for prediction, and you can see the prediction accuracy. Grain yield is this one, and you can see the. So what I am seeing here is that the simply inherited traits which have high heritability has given high prediction accuracy as compared to yield. I think we are further working on it to with new training population to refine the model and see that if we can further enhance accuracy for grain yield. Another approach we have taken is for enhancing genetic variability in the breeding program. If you see the breeding, it has to be accumulation of favorable genes, and as much as possible, we should use, the good by good crosses, bringing diverse sources, but see that, we accumulate favorable genes. And one approach is this magic, your multi-parent advanced generation intercross. So you select a set of, good high-yielding genotypes, could be varieties, popular varieties, see that they have matching phenology and cross them in all possible combinations. And then you can have this funnel crossing to double up 8-way crosses if you have 8 parents. And then you can, use it either for discovery or you can use directly in the breeding program also. So we, by single seed descent, we developed some line magic lines which will be used for discovery. But from the breeding point of view, I want to emphasize that, it gives you a lot of material. If you use the bulk which is available from 8-way crosses and take directly them breeding program, we have a lot of recombinations, and we have selected many lines which are giving higher yield than the best parent. So, and these are now being used in our breeding program. Some of the development in resistance to diseases, I will say that Fusarium wilt is one disease which was the most important disease, but one disease where we have made excellent progress, the breeding programs, because we have very good screening technique and very good sources of resistance. Interestingly, the resistance is highly stable. You can see this check, which is generally used, WR315, 40 years old, more than that, and is still standing there. So the resistance has been very stable for Fusarium wilt. And this, simple screening made it possible that now all the varieties being released, they have, high level of resistance to Fusarium wilt. Ascochyta blight is another disease which was very important earlier when large chickpea area was there in Punjab, Haryana, Himachal Pradesh, in that regions, and some part of Uttar Pradesh, Uttarakhand. And this is a foliar disease which is not as easy as Fusarium wilt. You don't get that high level of resistance. And if you take 1 to 9 scale, maybe you can aim for 3 score of resistance. But I will say that in recent years, we have made very good progress. And we have, a screening method here at Acreset in controlled condition, because the disease doesn't come here because of, we don't have that high humidity and low temperature. So you can have that. And then, we screen in Punjab, and this is from Ethiopia, the same material we sent there, and you can see some of the lines developed for Ascochyta blight resistance. Also, you can use markers. There we attempted. There are markers available for Ascochyta blight and Fusarium wilt, and you can see clearly that you have to see the efficiency, and you have to also see whether these are available in combination with others, or if you only Fusarium wilt, maybe you as well just want to screen in the field. But you have options there, and there is a variety actually released through marker-assisted breeding, or I will say marker-assisted back crossing in Karnataka. University of Agriculture Sciences Raichur, they developed this variety, and this is very interesting that earlier Annagiri 1 was the most popular variety in Karnataka and Andhra Pradesh. In Andhra Pradesh, totally replaced. In Karnataka also it is being replaced now. But this variety was one time a mega variety. And here in this case, markers assisted back crossing, if you want to recover the genome of that variety, if you want to retain other traits and only introduce Fusarium wilt resistant, so MABC is is one options. And then there is a variety that has been released, Super Annagiri, which is similar to Annagiri but has 7% higher yield and then resistance to Fusarium wilt. Resistance to part borer, of course, in the cultivated germplasm, we don't have very high level of resistance available. So efforts are being made to use wild species because there is slightly higher level of resistance available. But I think the ultimate, resistance or higher level of resistance we can expect from transgenics, and a lot of work is going on at many institutes, Indian Institute of Pulses Research, Kanpur, at ICRISAT also. And we hope that in the future you will have transgenics available for part borer also in case of chickpea and also in pigeon pea. And this is one priority trait which has emerged in recent years, considering the labor shortage and considering the mechanization of agriculture. We have to progress. We need to enhance mechanization in agriculture, and otherwise the farmers will leave the crops which are too much labor intensive. They will go for the crop which are can be mechanized. So for chickpea, if we want to make it competitive with other crops, we have to develop varieties which are machine harvestable. That they will, reduce labor cost and reduce the cost of production and increase the profitability. And another advantage is that at the harvest, you can bring the crop very quickly home, and avoid the damages that can occur during the harvesting. And of course, reduce drudgery on the farm women. And other advantages are also there in spraying, for the insecticides and light penetrations. These kind of plant types will be very useful. In fact, chickpea is harvested by machine only in developed countries like, Australia, Canada, US, and Europe. We never earlier paid attention. Otherwise, it's not very difficult to develop varieties, machine harvestable varieties. In recent years, we have, paid a lot of emphasis. And there are, in fact, you can see 2018 and 19 years, varieties have been released, 2 from Central Variety Release Committee, and others are from state, which are not only machine harvestable, but they even gave higher yield than the standard varieties. So this is, you have no yield penalty. The only thing you have to take it that you may have to increase a little bit seed rate in this case, because these are, erect type plants. And now, several machine harvestable varieties are in pipeline, and my assumption is that in the future, chickpea will be largely harvested by machine in India, just like other developed countries. Another area which is important is herbicide tolerance. If you're growing chickpea in irrigated conditions, or even in many other environments where weeds is the problem, you need herbicide tolerance so that you can manage weeds by spray herbicides. There is good variations we have identified, some lines we have identified, and this is one area which is emerging, and we are hoping that we will have in near future varieties which can be herbicide weeds can be controlled by post emergence herbicide spray. Also, one of our PhD students worked on mapping, QTL for herbicide tolerance, and we had this RIL population. And as expected, because usually in many crops herbicide tolerance is a single gene trait, we got one measure and a QTL for herbicide tolerance. Coming to market preferred traits, all the breeding programs in chickpea have been considering these, and I must say that chickpea breeding programs in India have done excellent work in considering the market preferred grain quality. These are the earlier varieties of Desi type chickpea. Now you have most of these kind of varieties in Desi type. In Kavli types, if you see in 80s we had a small seeded Kavli types. Then in the era came in Kak2 and other varieties came with large seed size. And in recent years, when we were importing extra large Kavli seed from other countries, we started breeding program in India, and now we have many varieties in this category. And in fact, development of good varieties, high yielding varieties in Kavli type, the Indian Kavli chickpea production increased, and we actually now became exporter of Kavli chickpea instead of earlier we were importer of Kavli chickpea. Now we are exporting. And the last trait which I want to touch on, where we have not done that much work, is further improving nutritional quality. Chickpeas are already nutritionally rich, high protein and high minerals, but we have a lot of variability. And there are opportunities that we can further enhance protein content, we can further enhance iron and zinc content. You can see the kind of variability you have. So this is one area where the breeding programs in coming future have to pay more attention. I was happy to I was listening to Dr. Tara Satyavati, and how in pearl millet these are now in mainstream, iron and high zinc trait. And there are minimum standard for the release of varieties, because the work started long back in pearl millet, and there is a good progress, good breeding material is available, and the program is at the is ready for that mainstreaming. We are not there, but I'm hoping that in chickpea also in near future, maybe 5 years down the road, or maybe 10 years down the road, these traits will be in the mainstream of the breeding program, just like pearl millet. Of course, because we have to, handle these traits in the breeding program, always parallelly we worked on, understanding genetics, identifying genes, QTLs for these traits. For protein, we developed, one RIL, and which we had a shared student with the University of Saskatchewan in Canada, and the student was able to identify one major, QTL, which accounts for 44% variations of protein and other characters. So these again have to be verified and used in breeding program. Now after that, briefly I touch on how can we further enhance efficiency of the breeding program. And this webinar was all about how we use biotechnological tools, integrate them in breeding programs to enhance efficiency of the breeding program. So if you see among all the breeders or the students of breeding, they know this equation, quantitative genetic equation, response to selection or genetic gain. These are the parameters. You have the genetic variability, which is there, important, then selection intensity, and then selection accuracy. So these are the things we have to enhance if you want to enhance genetic gain. And if you are calculating genetic gain over time, then how many generations we are taking per year is also important. If you are taking one generation per year, and instead of that, if you can take two, T will be 0.5, so you can double it. So you have to reduce the T, and you have to enhance this. And for these, there are ways for enhancing efficiency of the breeding program. Say, for example, taking intensity and accuracy. Intensity is directly related to the size of the breeding program. Means if you want to enhance intensity, you have to increase the size of your program. Means you have to evaluate more material. Then only you can enhance intensity. So for that, if you want to increase the size of your program, you have to go for mechanization and automations. You can't just do manual sowing and manual operations. So the mechanization and automation of breeding program is must to improve efficiency, from sowing, from harvesting, seed counting. All these have to be mechanized in the breeding program. The next is high throughput phenotyping. When you have a lot of material, you need to have high throughput phenotyping and with precision, so that you can evaluate large material and collect the data for that. Say this is high throughput phenotyping platform, which is totally automated. There is a 3D camera, and you can take many observations together. And there are many other approaches. Dr. Tara Satyavati was talking about XRF for iron and zinc. Such an efficient method. You can evaluate a large number of samples. Cost is low, and still accuracy is high. So you need to have not only for diseases, for many traits, all the traits, precision in phenotyping is important. Precision of field at trials also. The uniformity of field, the proper design, the number of replications, multi-location evaluation, all those things have to be taken care to improve again precision, and improve the accuracy. And of course, this webinar had a lot of emphasis, high emphasis, actually main emphasis on marker state selection and genomic selections. And these, because if you say marker state selections, here instead of seeing the effect of alleles, you're directly selecting for that. So you are avoiding environmental effects. So your accuracy goes high. And similarly for genomic selections, because you are calculating genomic estimated breeding values, and if you have very high correlation with true breeding values, then this is where you are improving R, actually selection accuracy, which is a square root of heritability. So if you have that equation with high precision, you are improving R there. So these things, marker stud selection, genomic selections, they increase accuracy. In some cases where the cost is low, even they can help you in increasing selection intensity also. So they need to be incorporated in breeding program. When you are generating a lot of data, then you should have digital data capture, the old notebooks, we have to get rid of them. We have to collect by using notepads, electronic data capture, directly load on the system, and very efficient data management system for the breeding program. So these are must for modern breeding programs. And then this we can't forget that all your modern tools and techniques will be useless unless you have the desired variability in your populations. If you don't have, then the markers will not work, your phenotyping will not work, nothing will work. So first thing is this, the variability for, and that too for the desired traits, the traits which you want to improve. Say if your target is the grain yield, you have to have the genes which contribute favorably to grain yield. So for that, choice of parents, how you use the parents in the crossing program, and there you will get help from all these. You have a good data management system where that will help you in selection of parents for your crossing. And the crossing scheme, as I gave example of magic crosses, so that you can enhance recombinations. So all these are very important. They are the prior requirement for developing genetic variability. Then you can move for these. So at ICRISAT recently, we have developed a rapid generation advancement protocol for chickpea, and which is directly addressing T. As I said earlier, you have to reduce T. If you are taking one generation, T equals to 1. If you are taking two generation, T equals to 0.5. You can take 4, T equals to 0.25. So you have to increase that. And here in this case, in chickpea, we have a standardized protocol. We can take 6 to 7 generation in a year. You can see this is, and what we have done here is manipulated two things, growing the seeds, immature seed, as early as possible, and then growing the plants with extended photo period so that you get early flowering. You combine these two, you can have a crop, a cycle completed in 50 to 60 days. And this is how you can reduce the breeding cycle, and which will, directly enhance your genetic gains or response to selection in the breeding program. So I think, these are the things I wanted to emphasize, that, we need to enhance efficiency of the breeding program using integrated approaches, whatever is available that can help in enhancing efficiency. So concluding, I must say for chickpea, I see clearly a lot of opportunities still exist for enhancing growth of chickpea production. By enhancing productivity per se, by reducing yield gaps, a lot of yield gaps exist. So means enhancing adoption of technologies, improved varieties and production technologies, and expansion of area also in rice fallows particularly. Then in recent years, as I explained, there has been good progress in development of, developing breeding materials for heat tolerance and suitability to machine harvesting. And I believe that these traits are now ready to be mainstreamed in the breeding program. Means we can decide that these would be must-have traits in our breeding program. Means all the varieties we develop should have these traits. And we need to enhance our efforts in the areas where we have done less work, enhancing, protein and micronutrient contents, and herbicide tolerance. And I'm sure these traits also can be in later on when we have enough breeding material can be mainstreamed in the breeding program. And for enhancing efficiency of the crop improvement program, chickpea improvement program, we must integrate novel tools and techniques, just like genomic selections, precision phenotyping, rapid generation advancement. These must be integrated. We need to, develop varieties as rapidly as possible, and because considering the requirement, of the farmers, of the industries, of the consumers, we need to provide them varieties as quickly as possible. With that, I thank you very much.
Thank you, sir. It was very informative talk, and we learned a lot from your talk, sir. Now I will pick some of the questions, sir, and I will request you to answer them. Sir, the question will be visible in your screen.
Well, I think in the management practices, it depends, which management practices we are talking about. Oxalic acids and these can be washed when, we have irrigations, waters. Otherwise, there is not much effects of other, management practices. Okay, next question is magic population for the same purpose in other crops also. Magic populations can be developed in any crop, but these are for the traits which are, controlled by many genes, polygenic traits, any trait like grain yield, maybe in some cases, protein content, which are governed by many genes where you can accumulate favorable genes, we can use this. The effect of silicon chickpea in increasing salinity tolerance, well, I didn't cover that, and this was not my area. We can skip that. Which race number of Fusarium axisporum are utilizing for screening for wilt disease resistance cultivars? The one example which I gave was actually the race 4, which was in progress, but there are markers available for other races also. Is there any chickpea variety improved through gene pyramiding for wilt and ascochyta blight? Well, by classical, approach earlier, we have, developed many breeding lines which have combined resistance to ascochyta blight and fusarium wilt. Very high level of resistance for both, even including botrytis gray mold resistance. Is it possible to grow chickpea around the year with background of heat tolerance with acceptable yield? No, it's not possible. At least in rainy season, we can't grow chickpea. It can be grown only in dry areas, dry season only. So you can grow in the winter and maybe a little bit in spring and summer. What is the range of zinc and iron content in chickpea seeds? I think I gave provided one slide which had the details on that. Generally the current varieties are in the range of 20 to 30 and 40 ranges, but we can almost double these, or maybe at least can have 60-70% higher fees. Yes, please. Next.
Okay, thank you so much, sir. Thank
Okay.
you so much. It was very informative session, sir. So now go to the sir, now we will conclude our 2 days national webinar, and now we will go for validatory online validatory session, sir. So the chief guest for the online validatory session is Dr. C.M. Gaur, sir, Director Asia Research Program, University of Hyderabad, and chairman is Dr. Professor S.K. Rao, sir, our honorable vice chancellor. And for expert comments, later on I will invite God sir, and for this special remark, I will invite Professor S.K. Rao sir. So, sir, I am compiling the session, sir. So, the 2 days national webinar on application of biotechnological tools for crop improvement has been organized by RVSAVV Gwalior on 10th and 11th June, sir. Dr. Nagendra Kumar Singh, National Professor and Director NIPV New Delhi, was chief guest for inaugural session, and it was chaired by our honorable VC sir, S. K. Rao sir, and MP Jain, Director of Research Services at RVS KVP Gwalior. MP Janser has welcomed the chief guests and participants in the welcome address. And after that, keynote address was given by Professor N.K. Singh sir, and he mentioned that biotechnology is relevant area and various tools of biotechnology are available for crop improvement. And biotechnology is, as it is a tool, it should be used with all the crop improvement programs to get it in less cost and in less time set. Then our Vice Chancellor in his presidential address, he told that biotechnology need to get as integrated approach and it should be used, he pointed out we should have knowledge of all the subjects, then only we can get improved variety in less time and for particular type of interest. Sir, the first day, and that was 10th June, the first lecture was given by Dr. Nagendra Kumar Singh sir, and on decoding of plant genome and its use in gene discovery and varietal improvement. He told about rice as he has been involved in sequencing of rice, wheat, and tomato genome, which was international project. So he told briefly about these genome projects, and then he was also involved in jute, mango, and pigeon genome sequencing, where he lead the that crops and sequencing was done in India itself. Then he told about all the possible outcome, means how why we go for genome sequencing and what is its applicability in crop improvement. So session was very interactive, sir, and he answered several questions of the participants. The second lecture was given by Dr. C. Bharadwaj sir, who is Principal Scientist at Division of Genetics, ICR, IRI, New Delhi. And his work was on crop improvement for stress tolerance using genomics assisted breeding. Sir, he compiled several aspects for crop improvement. He quickly told about chickpea improvement, also QTL mapping, about genomic selection, about 5G approach for genetic gain and improvement of crops. So the session was very interactive and he told all the possible aspects of biotechnological tools for improvement of these crops. Then the third lecture was given by Dr. Amita Mitra, scientist, ICR, MIT, New Delhi, on genesis and principles of QTL mapping. Amita madam has very nicely delivered her talk and she explained conventional methods of QTL mapping as well as single marker analysis, interval mapping, everything she explained very well and told the importance of molecular marker in QTL mapping and how it will be beneficial for crop improvement in using molecular breeding approaches. Then afternoon session was there were two lectures, one given by Dr. S.R. Malu sir, who is former Dean MPUAT Udaipur, on accomplishment challenges and strategies for sustainable agriculture. He told about the climate smart agriculture, how we can go for climate smart crop, why we have, what is important of climate smart crops, and how we can use biotechnological tools to get these climate smart crops. After that, Dr. Rakesh Srivastava, who is principal scientist at ICRISAT, sir, he told about pearl millet genomics and molecular breeding. He also described about QTL mapping approaches, genomic selection, as well as using SNP chips for getting that high throughput genotyping and molecular breeding approaches. On today's the technical session was chaired by Dr. A.K. Singh sir, and the first presentation was given by Dr. Maheshwari Madam. She is Professor and Head Division of Crop Sciences, Frida, Hyderabad, and she delivered her talk on phenomic approaches for enhancing abiotic stress tolerance in grain-fed crops. She told the phenomic facility which she developed at Frida, Hyderabad, and tell all the important aspects of why phenomic facility is important for crop improvement, how we can get high quality data by using phenomic facility. It was very informative session and the students interacted with her and asked lot of questions regarding phenomic facility and its infrared lights, etc. Then second talk was given by Dr. Sharad Tiwari sir, who is head at Genetics and Plant Breeding Department, JNKVB Jabalpur, on molecular techniques for identification of species and genotypes. Very nicely, sir delivered the talk and told about DNA barcoding technique, how they have used DNA barcoding for barcoding of millet, soya bean, etc. And he explained all the steps and techniques, and his high quality paper using that technique. So that lecture was very informative. And afternoon session started. First lecture was given by Dr. C. Tara Satyavati. She is project coordinator at Tripon Pearl Millet, Jodhpur, on high iron pearl millet in context of biofortification in crop plants. So, madam, very nicely, madam explained about whatever work is going on in pearl millet. Means in single lecture we could know whole story of pearl millet, why it is means where it is lacking, where we have to go. So biofortification is one of the important aspect of pearl millet breeding, and she told about iron zinc content and how we can improve the pearl millet for iron and zinc content. So last lecture was given by Sir Dr. P.M. Gaur, Director Asia Research Program, Research Hyderabad, in advancement in chickpea improvements topic. So lecture was very informative, very easy deliverable, means everyone can understand, very easy to explain. So sir told about breeding of drought and heat tolerance in chickpea, as well as genomic selection and QTL mapping for drought, heat, as well as fusarium build also. Sir told about molecular breeding aspects and using that SNP chips also for high throughput genotyping, and he emphasizes on use of biotechnological tools to release variety in less time. So that's all. And I want to clarify one thing here also. Sir, in our webinar, around 2,000 candidates get registered from all over the India, even from Nepal, Iran, and all the nearby countries also. I don't know how link reached to them, but out of them, 75% were students, 25% faculty members, and from several universities, sir, RBS KVB, JNKVB, BHU, TNAU, Pantnagar, Bundelkhand, and etc. All the university, even IRI persons also. So we are very happy with the response. Means out of them, 13% were UG student, 35% PG student, and 51% PhD student was there in that webinar. And 42% female candidate, 58% male candidate, so almost equal. Now today we have given them feedback link. So around 500 feedback link has means come back for the from that participants. So it was really great experience, sir, and first time we have done that work. So that's why a lot of technical issues also coming between. But like that way we are learning actually. Yesterday also we lost the link, then we resend means new link to them. So it was really new learning for all of us. But anyhow, in that crucial time of that COVID, we have utilized that time and listened all the great lectures by eminent scientists. So which is very for that we are very thankful to all of the speakers and organizers and our honorable vice chancellor, sir. Now I will invite Dr. PM Gaur sir for his expert comment.
Thanks, Dr. Tiwari, and I must appreciate that this is really was very successful webinar. I was also yesterday, because I was busy in another meeting. I was in the middle. I was going to it, and it was so simple to just see it, and the participation is amazing. We could reach to so many people, and this is our next generation of scientists, and definitely they will inspire, they will with some of these things. As Dr. Rao said in the morning yesterday, this is the crop breeding. When we talk of a breeding program, is integrations, integrations of all tools and techniques. For a plant breeder, if you even take the discipline, for us, all disciplines are important. We need pathologists, we need entomologists, we need agronomists, we need quality people, we need all of them. Similarly, in technology for the breeding programs. For us, all those technologies which can enhance efficiency of our breeding programs are important. Now, if you see the biotechnological, technologies, evolution of technologies, initial from, the transgenics, such a great tool, such an, you have tool in your hand where you can bring any gene from any source, the kind of variability you can bring. Such a powerful tool, though, of course, we couldn't, really utilize it much because of the regulations. But, if you still see the transgenics, which have been developed and grown globally, you have more than 190 million hectare area grown globally on transgenics. And even in India, the BT cotton, we are in transgenics, we are fifth, largest transgenic crop growing countries. The kind of impact, these have, because, we could never imagine. We were always looking for genes in the cultivated germplasm or wild species that we can bring, gene from other species, other organisms, and, enhance our efficiency. So that was, really a great in case of transgenics. Of course, the difficulties in regulations, and we could not really truly exploit the potential of that was available for transgenics. Now, the other, field that has come up, is genome editing, where you can still modify, the variability which is not existed. You can, change them at desired locations and bring the change. And of course, we will see, again, emerging new technologies coming up. Now, there are other techniques, we have to enhance efficiency. As I said, again and again, I have been saying that in crop improvement program, whatever it can enhance efficiency, molecular markers, they give us power for selections where, you can have increase. If you see that breeder's equations, which I showed, where you have to increase intensity and accuracy, both these things are handled very well when you can use markers in the breeding program. Now, you have to decide where you should use what. You can be, as Dr. N.K. Singh yesterday, he was giving very good presentations, and he gave one example of, the plant height markers. Anybody can ask why you need a marker for plant height. You can just see it. And you can measure it with a scale or just see it. You don't need marker for that. Now, you have to make a decision where you have high efficiency, and where you need, or whether it is available with combination with others as a bonus, you can use it. But if you have markers, like say, for example, for only one trait, you have to make a decision whether it is going to enhance your efficiency or not. Either it has to reduce time or save money or improve efficiency. Now, I give example like many times that, say, for example, you have to travel from, Gwalior to Delhi. You have got different means. You can fly, you can go by train, you can go by road, and you have to make a decision, which is for you the best. May not be for others, that's the best. So the breeding programs have to see their own resources and the technology they have available with them. Tremendous potential is there, in use of markers. We have to have, more of them. We have to validate them. Many times, as Dr. Rao said yesterday, that, the QTLs are identified, papers are published, and we leave them there. This is true. In many cases, we just stop there. But if we want to use them in a breeding program, we have to take only the major effect QTLs. Forget about those minor QTLs. If somebody is saying, I got 10 QTLs for seed size, forget that. Leave it there. You just take the weight. So, but we can always integrate those major effect QTLs in our programs. And if you have many of those coming up, and you can develop the selections tools, which will increase your efficiency in the early generation selections, that will help you. I mean, as Dr. N.K. Singh gave example, MABC has been very, very successful. It's such an efficient method for, the breeding program, because a perfect case actually for the success of markers, where you want to have a mega variety, you want to retain all the traits, you want to improve one. You can have foreground selection, you can have background selection, nothing like markers for that. And that is why, you have, most of the varieties from markers have come from MABC, because that's the perfect case for use of markers there. You have to, we have to move further. The mega varieties may be disappearing now when you have these new varieties. The changes, like 10 years old varieties will not be grown. We have to move forward. If you use MABC, it's good for improving mega varieties, but you don't make genetic gain actually. Your yield levels are not much change. So we have to make genetic gain. We have to look for that. We have to bring new combination. We have to enhance yield potential and other things. So another important area which has been emphasized and should be taken care is phenotyping. We can never ignore, the advances which are required in phenotyping. There are many cases where simple, efficient phenotyping is good enough. Dr. Tara Satyavati gave example of XRF, like you have a choice. You can use markers, or if you have a very efficient method, like we are developing for a groundnut for a oleic acid, an IRS where single seed you can do it. So you have a choice. You can do it there. So, and then even for your discovery also, you need to have precision in phenotyping. Then you will have, your accuracy in other things also. So all these things are, important. Important thing is integrations. Beginning of the era when we started, biotechnology, we used to have separate institutes, because that was a need that time. We needed an emphasis, high emphasis in development of technologies and tools. Now this is the time of, integration. We should bring them together. We can't have, molecular breeding separate program and conventional breeding separate program. No way. We got to have one integrated program and integrate, all the technologies and tools available with us. And, there have been, very good changes in technology development. I remember, in the beginning earlier when we, started molecular breeding, and when the breeders wanted to have training in molecular breeding, and we used to go to the, for training, first thing they used to give is, you a quote and start, how you extract DNA, how you have a centrifuge, how it, the jails are run. So the plant breeder has gone there, but now we know well that, you don't need to know all these things. What you need to know is basic principles, and technologies. You can give your samples, even leaf samples directly to a service lab. They will provide you data. So you don't need to know that part. You should know your part very well. How efficiently you should use the data in your breeding program. How should you integrate the, different technologies in the breeding program? I think, the things are moving very fast, and the kind of advancements are taking place, as I gave example of, rapid generation technologies taking, 6 generations per year. Now they are going to enhance our efficiencies tremendously. And we need to do that. We need to reduce time for development of varieties, because, the farmers can't wait that long. The consumers can't wait that long. I think it was, I was really privileged to, join this and thoroughly enjoyed it. Unfortunately, I had missed some parts yesterday because of another meeting, but I was trying to, in the night, I saw them in some of the presentations, because I just went through them. So it's a very good platform. I'm sure, our students and other young researchers must have been benefited from it. And thank you very much, Dr. Rao, and all, Dr. Tripathi and Dr. Sushma Tiwari for this opportunity. I thoroughly enjoyed it. Thank you very much.
Now I will request Dr. Bharadwaj sir to give his expert views.
Good afternoon. I think it's almost into evening. I thank the organizers, particularly Dr. Rao sir, Sushma Tiwari, and all the group who have actually conceived this idea of having a webinar. In the COVID times, we proved one thing, particularly that for dissemination of technology, physical presence of togetherness is not required. And then literally we can reach a greater audience, uh, pan countries, pan regions at a quicker pace. That's true that the integration of technologies is the call of the day. We have technologies at hand, like yesterday I was saying. The breeder should judiciously use these technologies in the crop improvement program. No technology can be standalone, and no technology is definitely superior. For example, for better introgression of alleles and removing the what you call as linkage drag, perhaps the marker link QTLs would work great. Simple inherited traits like Dr. Gaur was putting, normal breeding technologies would work very effectively. So a breeder has to judiciously use, and we don't require sophisticated labs. We can use the center of excellence centers at ICRISAT, at IRI, and at other places, or a centralized lab that can cater to this. But what is most important is that a breeder is aware of these technologies and knows where to chip in this. So with the advance of technology, the improvement as well as breeding efficiency is going to increase. And certainly now each government or each university will be looking at what is the cost is to benefit ratio that actually comes from the breeders output. I thank again the organizers, sir, and also Dr. S.K. Rao, sir, for actually giving me an opportunity and for this excellent webinar. Thank you all.
Thank you, sir. I will request Tara ma'am to give her view.
Good evening, everyone. Thank you for giving me this opportunity to share my views about the webinar. As mentioned by the previous speakers, it was a very excellent national webinar reaching the corners, uh, every village and city, because the students are at their native places and they're taking advantage of technology, and you were successful in making them learn even in holidays. And also, as mentioned by Dr. Kaur, it is indeed the time to integrate all available technologies, including information technology, like what we are using now. Not only breeding in the field, breeding in the lab, breeding with the technology, many things we are integrating. And this is the right time and also the right platform for people to learn. And I think the students who are attending this national webinar yesterday and today might have with this. And the purpose of these things is to incite and ignite their minds so that they can think for the future and start dreaming about what they want to be. So you never know which thought will ignite the mind of a person. But definitely, such webinars are the platforms to ignite the young minds, which are going to be the future of this country, not only this country, in the entire world. As I mentioned in my lecture, and also you have seen from different lectures, we are the process of evolution. We are already integrating many things, and we wish that we'll go in a long way in achieving not only food security but nutritional security and also emotional security in this time. And this is one platform which is bringing everyone, binding all the emotions and bringing them everyone to. Thank you, sir, and also thank Dr. S.K. Rao, sir, for giving us this opportunity to share our research and also share our views. Thank you so much.
Thank you very much, ma'am. Now I will request our Honorable Vice Chancellor, sir, to give his special remark.
Good evening, friends, and it is a very good opportunity for all of us to understand about the latest advances in the different areas by the eminent experts. The experts like Dr. N.K. Singh, Bharadwaj, Amita, and Malusab, and Rakesh, and Maheshwari Madam, and Sharat Tiwari, and Tara Satyavati, and Dr. P.M. Gaur, and all these things, they have been a very good team leaders in the research, and they have delivered lectures in a very simplified form that any graduate student, even undergraduate students, can able to understand what it means, what they want to communicate. That is the beauty of this webinar, what I have perceived out of this, and a lot of clarity in the presentations and a very simplified form, the complex things also. Now, as I rightly said in the inaugural session, now this new generation plant breeders, they require this type of training, and then only they can deliver the products by reducing the yield gaps as well as breaking the yield barriers. As you know, now we are talking about, everybody talks about food security, and we are self-sufficient in food production, this and that. But if we really look at the other countries like Japan and other countries, in our country also, this is a temporary phase of food security. This is not a permanent. Why? Because we don't have a lot of land now. The land is limited, population is increasing, water is declining, and the soil health is declining. Everything is in the negative trend except the crop improvement program. The plant breeders are putting the efforts to improve the yielding varieties. And under this situation, the only agricultural scientists are the solution. The governments has to look at with the huge investments and better policies and better investments. Then only we can able to survive our future requirement. Our food prices are increasing, food requirement is increasing, but land is not increasing, productivity is not increasing. The food security crops like rice, wheat, and maize almost stagnated all over the world, including India. India is not an exception. But how to break the yield levels of the food security crops like wheat and rice and even maize? It is, I don't think any amenable solution for this unless you invest a lot of money in this area of crop improvement with the biotechnological tools and with good trained plant breeders. That is the only solution. If the once the good variety or hybrid comes, automatically it's agronomy and all these things, it follows. That is the only solution to meet the food requirement future. Otherwise, if the day India start importing food, that we have gone. Because it is the only country having the strong agricultural base with the diversified ecologies and diversified crops and diversified genetic resources in terms of the vegetables as well as in spices and even in the cereals also. No other country is having this much of diversity of the crops. And all these presentations have given a very good insight how the breeding programs are being run at the different institutions, and even the fresh plant breeders, they can able to understand what they have to do for their future breeding programs. With these few words, I thank all the experts, especially Dr. Tara and Bharadwaj and Gaur Saab and all the NK Singh Saab and even Rakesh from ICRISAT. Very nice presentations. They have also addressed the nutritional security for future requirements. Everybody talks about nutritional security nowadays, but very few centers they are working for biofortification and all these things. But their farmers are not getting the good price out of that. As Tara rightly said, that is not being valued, and the food value chain of this high value nutrient-rich foods are not being recognized in the value chain, and the prices are not being paid to the farmers. And that is the major limitation. There lot of policy decisions are required on that, and even the industries also has to recognize that thing with the nutrient-rich products. They should be able to pay the produce high value. Then only these systems will survive. Like QPM, maize has been developed by the CIMET, and nobody is purchasing at higher price. And unless it has to go to the PDA system and the government has to recognize it and contract farming and all, then only it will go. And industrialization also, the MSMSC industries in agricultural sector, all these can help to improve the utility of these nutritionally important products. That this is the requirement really. We are spending a lot of money on healthcare. If we can solve these problems with this with the dietary habits, then only we can save a lot of money and improve the nutritional health in the rural areas. Now, for the last 3 months, the government have made a lot of policy changes that is favoring the agricultural sector. Previously, nobody used to listen us. I have written many times to the state and even the central government also, the Union Agriculture Minister, I told him, sir, these are the requirements. Now, this COVID-19 has made get into their brains, and then they see a lot of changes have been made in the marketing of the agricultural produce of the farmers and input supply systems and subsidy patterns. And if now they realized it, if the farmers, if they cannot survive, we cannot survive. If they can't produce the food, then we cannot get the food. And if this sort of understanding is there now with the political leaders and the policy makers, now we can have a better platform for future, I think. I hope all the best for all of you for giving the nice presentations. Thank you very much for coming and giving the presentations. Thank you.
Thank you, Dr. Thank you very
Thank
much.
you very much, sir.
Thank you very much.
Now I will request our head of the department, Dr. M.K. Tripathi, sir, to give vote of thanks.
Thank you, Dr. Susuma. Good afternoon to everyone. I first thank to Great God for successful completion of this webinar. Application of artificial tools in crop improvement held between 10 to 11 June by the Department of Plant Molecular Biology and Biotechnology and Genetics and Plant Breeding and Genetics. In the super guidance of our teacher, guide, mentor, Honorable Vice Chancellor, sir, for his motivation, dedication, and sincerely presence during whole webinar. Then I thank to Professor and again Chief Guest and for their keynote address. He has delivered very impressive, updated, and wonderful presentation. I also thank you to Dr. Sri Bharadwaj sir. He is also my senior during my PG degree. They did PhD from the JNKVB Jabalpur, and they have given very experienced, applicable, and comprehensive speech. I again thank to Dr. Antamitra, Madam Scientist, NIPB, New Delhi for the energetic, applicable, and excellent presentation. Then I also thank to Dr. S.R. Malu sir, yes team, MAPUT, Udaipur for his vast experienced and remarkable presentation. I also thankful Dr. Rakesh Kumar Srivastava sir, Principal Scientist at Agriset Hyderabad for his energetic, impressive and knowledge about the pulmonary molecular breeding. I also thankful Dr. M. Maheshwari Madam, Professor and Head Division of Craft Science, Rita Hyderabad for innovative ideas about the pyramid facility which is the need of today for the climate resilient agriculture. And I also thankful to our advisor, MSc and PhD and my teacher, Dr. Sarat Tiwari sir, for his nice presentation on the bar coding of different crop species. I also thankful to Dr. Sitara Saktwati Madam, our project coordinator, because by present I am working it as a breeder in agriculture. She has delivered so beautiful lecture, and that is very useful for the plant breeder of the working on the pulmonary. And last, Dr. P.M. Gaur sir, from 1996, I heard them, sir. Sir taught me two courses. That was the cytogenetics and genetic engineering. After long time, I have heard Dr. Gaur, sir. In fact, I have not seen so simple teacher in our whole life. Even during school education, I studied in model multi-purpose school Diva, where the very simple and knowledgeable teacher, but so simple teacher I have not seen never in my life. I am thankful sir for invitation of our vice chancellor, Susma Madam, and give your valuable lecture. And now I am going to thankful to our organizing committee, Dr. Radha Villare Madam, Dean Faculty of Agriculture, and co-patron of our webinar, DRS Dr. M. P. Jain sir, for their uh, welcome address and guiding us. TF Dr. S. Upadhyay sir, DI Dr. A.K. Singh sir, and chairing the session, Dean Dr. J.P. Dixit sir, you with the also joint convener, and B.S. Khandelkar sir, professor and head forward of genetics and plant breeding and PMDNB. Dr. Raju Kahuja sir, Dr. Rara Sikarwar sir, Akhilesh Singh, Dr. N.S. Bhalodia sir, for as working as a reporter, and last, Dr. Sushma Tiwari, the organizing secretary of our webinar. She has done a lot of work, so very sincerity, from deep, and why her effort this can be possible. And at last, I grateful to Mr. Vinod Patel, technical for technical support, and Mr. Uh, Gyanchand. You are helping us for the whole webcasting and browser participation. Or at the final, I thank to our participants belonging to various research institute, universities, public and private sectors of different disciplines. Without them, there was no hope to competition of such webinar. And as mentioned by Dr. Gaur sir, Madam Dhara Sapswati, Dr. Bharadwaj sir, and Dr. M.K. Singh sir, this is very few part of biotechnology has been covered so far. So our intention to organize an international webinar on if lockdown sustained for 1 month more, and I hope all you will help us. Thank you very much, sir. And I forget to someone to mention here, please conclude yourself in this webinar. Thank you very much. Thank you, sir. Thank you, madam.
Thank you.
Thank
Thank you, sir.
Thank you, madam.
you very much. Thank you.
Yes, sir.
Thank you, sir. Namaskar.
Namaskar. Namaskar.